Okay, so people is coming. I'm sure of that. Um, this is this is our third and last day of workshop. The volcano has treated us well, so we can complain. And today we will start our third session called Circular Economy and Sustainability. And this will be followed by Scientific Entrepreneurship in the afternoon. So for that, I present Dr. Maracea and Dr. Carolina Merino as co-chairs.
No? No. Morning. Today, the first speaker is Ekaterina Filimonenko from the Institute of Environmental and Agricultural Biology from the University of, sorry, my pronunciation, Tumen? Tumen. Okay, welcome, Ekaterina. You can start.
Good morning. Today I will share with you the results of our project, of our two projects, Terra Arctic and Carbarus, where we studied, how they change soil organic matter parameters under the fire in the tundra environmental. Every day, thousands of fires, small and large, are happening globally. And for high latitude is very important because the fire is a marker of climate change. And under the climate change, temperature getting higher, humidity is go down, and also vegetation has a dry state. And all of these factors form the fire weather conditions, and as a result, the fires start to be more frequently, burn intensely, and spread to wild area. And it's also true, and especially true for the territory in the north, including the tundra territory. And we work in this area. And for the north of Western Siberia, the second important factor for the fire is the land use, because the north of our region is the most important area in the Russia where oil and gas production. As you know, when oil and gas product, any technologies every time includes a lot of churches where waste gases from the crude oil are burned, and under the fire weather conditions and the huge amount of church, it could lead to the fire goes to the tundra, and a lot of area in the tundra are burned as a result of climate change, and in combination with the use of territory for oil production. And after the intensive beginning of oil production in tundra in the middle of the 19th, it led to the very high increased amount of burnt area in the tundra. And nowadays, more than 10% of territory of northern tundra are burnt, affected by the fire for the least 35 years. And while fire have huge numbers of impact of very different part of our life, including economics effects, social effects, and of course effects on environmental, including destroy vegetation and wildlife, and also as a result of fire, water and air polluted, and especially fires leads to decrease the soil quality. And the most important to study how fires affects on tundra soil, because in tundra soil a lot of amount carbon is stored, and its amount could be compared with another type of ecosystem. But the special features of tundra is that after fire, vegetation and litter are destroyed, and it leads that surface gets more darker, and it decrease the albedo of surface, and more solar heat fluxes go to the soil, and the active layer through layer of the soil get bigger, and it could change the biochemical processes in the soils, and really strong change the biochemical processes and processes with soil organic matter. And effect of fire on the soil properties is very complex. So fire's effect on the chemical, physical, biological properties. And for our project, we focused on the several parameters of the soils. Most of them is related with the soil organic matter, microbial biomass, microbial respiration, and also some other parameters, including changes in some cycle of chemicals. So under the fire, a little layer is very strong decrease in the cellular times. And it leads to changes in the vegetation type of tundra, because when the active layer goes bigger, it leads that some forest could appear in the tundra, and in some cases, after 10, 15 years, really forest could appear for the old burned territories. It's really changed the processes in tundra. And according to our results, our studies, soil organic carbon really strong control the one of the most important parameter about water in tundra, water holding capacity. And under the decreasing of content of soil organic carbon in the tundra after the fire, it leads also to decrease of water holding capacity. For the liter horizon, magnitude of decreasing is around 2 times. The content of soil organic carbon decrease in the liter horizon in 10 times higher as usual than for mineral horizon. And after the fire, the content of carbon is decreased up to 40% for any types of fire what we studied. And what about the content of total nitrogen? Is decreased in different magnitude for liter and for mineral horizon. For liter horizon, it's around 13%, and the more huge amount of decreasing was found for the mineral horizon. It's around 40%. And also the ratio of carbon to nitrogen is also decreased under the fire. And the most significant decreasing was found for mineral horizon because of differences in amount of decreasing carbon and nitrogen, and also because of processes of leaching when the mineral component, including nitrogen component, leach moves from the burned liter to the down and accumulate in the mineral soil, topsoil. And soil pH for tundra soils range from around 4 to around 6, and it's typical value for the tundra soil. But under the fire, this value is really strong change. It's getting bigger for the mineral horizon, in some cases up to the plus 1.5 units. It's because two processes. First of all, because ash productions and with the mineral component, and another reason is organic acids from the organic matter is decomposed under the fire, under the burning. For the studying how fire changed the stability of soil organic matter pools, we have applied method of thermogravimetry analysis, and we can see very clearly separate the peaks related with labile soil organic matter and the stable soil organic matter pools. Under the heating soil samples from the temperature from the 30 to the 600, the more and more stable soil organic matter compounds start to be burned and go out from the sample. And we can see the pool of labile soil organic matter is bigger than the stable, and under fire leads to the decrease the pool of labile, and after the next time during the post-fire regeneration, the volume of the labile pool is recovered. And energy, what is released as a result of burning soil organic matter, is very strongly related with temperature and with energy for this analysis. And according to the maximum of these two peaks, we can say that, that the stability of stable pool of organic matter is 1.2 times higher than the stability for the malleable pool. And the thermal energy, thermal effect from the burning of soil organic matter is decreased under the fire for liter horizon up to 1.3 times and more than 3 times for the mineral horizon. Fire have a very strong effect on the microbial biomass carbon. In the shortest time after the fire, carbon of microbial biomass is very strongly decreased due to the burning. But several years ago, the carbon biomass is quite close to the unburned level, and then for the next time, during the post-fire regeneration, post-fire recovery, this go to overtake the unburned level because of more vegetation produced for this burned territory. The same processes The same processes we can find for the mineral topsoils, but there are time lag because of fire in the tundra is very fast and it's not enough time to affect for the more deeper layer. But the same situation with the same processes that after more longer time after fire, the recovery processes leads to the microbial biomass for the burned area start to be bigger than for control territory. So fires also decrease the microbial respiration for both horizon, for liter, for organic horizon, and for mineral horizon. And the magnitude of this decreasing for the liter is around 30-35%, and in some cases for the mineral horizon, it could be around 50% of parameters of microbial respiration. And if we will talk about specific respiration, that was estimated as a ratio of microbial biomass respiration to the content of soil organic carbon, we can see that fire leads to the really strong decrease of this parameter for the most cases. And it could reflect it that after the fire, soil organic matter start to be more sensitive to the microbial decomposition. And it was found that the shortest time after the fire, the soil organic matter is more sensitive to the warming because of parameters of temperature sensitive of soil organic matter to decomposition is get bigger for the burnt areas, in some cases up to 0.2 units. So another point is very strongly related with the parameters of changes in soil organic matter under the fire is a change in the fate of some chemicals. It's especially important for the chemicals that could be volatile under the burning, such as arsenicum, mercury, some radioactive component. And now we have studied what type of changes there is with mercury, because it's very dangerous chemicals because of their toxicity, and it's very ancient known elements. It was used for many technologies processes in the past, especially in the 20th century, and of course the most previous period for gold mining, for some technical measurement and so on. And the most useful, it was the 20th century. And as I say, the huge problem of the mercury is its toxicity effect on the human and all the life. And after some cases in the world when mercury have a very strong destroyed effect on the health, the Organization of United Nations was prepared the document to compare between the government to the decrease effect of mercury on human to protect human and environmental from the emission of anthropogenic mercury from any sources. And it's really lead to the uses of mercury was really decreased in some decades, and now mercury is the only one chemicals which have a deeper technophysical compared with another chemicals, because most chemicals continue to be very active use in our life for the new technology, for the new processes, but now mankind, humankind very clear understands the dangers of mercury, and it goes down. And we can really understand that for the 2 or 3, maybe decades last, but for the previous 2 centuries, we released a huge amount of the mercury in the environment, into the atmosphere, hydrosphere, soils, and so on. And we can solve problem just for 10 years. And now the problem of mercury is also still with us, and a huge amount of mercury goes around the geospheres. And there are only 2 real ways how we can remove the mercury from the cycle and store it. It's stored in the deeper ocean and stored in the deeper horizon of soil. And all these 2 ways takes time, needs time for these processes. It's really longer processes. And when mercury deposited on the soil, time by time it could be go down, but there are some processes which return mercury, release mercury back to the atmosphere, and one of the most of them that is biomass burning. And so that's why it's very important to study fires in the point of how its effect on the chemicals mercury. So for the deposited mercury in the soil, there are 3 ways, 3 main ways. From the dry deposition in link with dust, with the wet deposition, with the water, snow, rain, and so on. And also mercury can go to the soil through to the plants, because when plants grow, they could uptake mercury from the atmosphere and enter to the soils through to the organic components. And fluxes from the soil and atmosphere have 2 directions, from the soil and also to the soil. And plants is very important component for these fluxes. And these processes can be controlled for many factors, including physical, chemicals parameters, and also, of course, meteorological parameters, surface, soil surfaces parameters. But the most important of these parameters is organic matter, because more than 90% of mercury in the soil is related with organic matter. And if we will see how mercury distribution on the surface soil layer, we can see that tundra soils contain around 8% of the deposited mercury on the surface. But this is only for the up layer of the soil. But if we will see how mercury distribute for the more thicker layer, up to 1 meter or up to 3 meters, we can see that for the North Hemisphere, the permafrost zone is the main reservoir for the mercury. And the content of mercury in the permafrost is 2 times higher than for the other type of soil, ocean, and the atmosphere. And the very important thing that the mercury in the permafrost is very stable. It's removed from the biochemical cycle, it's just stored. And the changes in the permafrost state could lead to the elevation problem for the mercury cycling. And it's also a point where we have studied how the mercury effect on the fire. And for the tundra, forest tundra, we can find that the mercury content is very strong controlled by the organic carbon and the total nitrogen. And also the content of mercury for the littoral horizon, for the organic horizon, in 4 times higher than the average mercury content in the Earth crust. In the same time, for the mineral topsoil, the content of mercury is equal to the Earth's crust. It means the reason why mercury is in little horizon, its uptake from the atmosphere, it's not geological factors. And the tundra ecosystem can accumulate mercury from the atmosphere and store deeper. And after under the fire, content of mercury really decreased for the little horizon. In some cases, up to 35%, 36%. For the mineral horizon, it's not so significant, but in some cases it could be. As you remember, decreasing of carbon under the fire is around 40%, and little bit bigger than for mercury. It means that mercury content, it's also controlled in the soil by content of iron, sulfur components, and the clay minerals, but of course mostly by the organic carbon. As I say, the fire really decreased the deeper of the layer where the freezing zone started, and after the fire, this layer goes down, in some cases up to 2 times like this, and it means that the more content of mercury and other companies, geochemical companies, could involved in the geochemical processes. But of course, fire mostly affected on the upper layer, when the decreasing mercury under the fires could be more than 3 times and so on. But also, for the deeper, it's also there is effect of the fire, the mercury content, and after the fire, mercury goes to the atmosphere and continue to cycling. During the post-fire recovery, when the vegetation ups to bigger, stronger, mercury content is also get bigger in the litter because of new vegetation. Take mercury from the atmosphere and continue the processes of store it in the layer. So, for the tundra ecosystem, during our project, we was found that the carbon, nitrogen, and the ratio is decreased under the fire. Thermolabile soil organic matter and thermal effect of the soil organic matter is also decreased under the fire. Microbial biomass pool and microbial respiration, water holding capacity, and some other parameters is also strongly decreased under fire in tundra. And pH soil is increased under the fire. And soil organic matter is very strong controlled for some chemicals, including the root, mercury. So, thank you. Thank you very much for your attention.
Thank you, Ekaterina. We have time for some questions. Marilu? No? First Marilu, and later Francisco.
Okay, thanks, very nice talk. My question is related to the percentage, the organic matter the organic matter percentage after fire. You say 40%?
Yes, the magnitude of decreasing of soil organic carbon is around 40% from the litter and the 40% from the mineral horizon in the shortest time after the fire.
40% of what is in value?
Sorry.
The value of
We can try find it. Yes, this I can.
yes, but what is the initial value?
Initial value, it's before fire, the content of carbon was around 35%. In the liter horizon, it's very high. It's a liter.
Okay.
And for the mineral horizon, it's around 4%.
Yeah, because I am very surprised. Thank you very much.
Thank you very much for your presentation. I don't know if it's here. I have two questions. One is the mercury in the soil is accumulated by the oil industry, or what is the sources of the mercury?
No. Oil industry is the reason why fire is appear in the tundra. It's not a really strong reason why mercury released into the atmosphere. Of course, some content of mercury could contain in the crude oil. And under the production, under the produce oil, gas, it could be some part of them go to the atmosphere. But the most reason why mercury is in this North ecosystem is the global processes. It's not local emission sources. It's global circulation, and mercury from another part of the globe go to the atmosphere fluxes in the north and south latitudes.
Thank you. And my second question, sorry, is the effect in the permafrost is, you know, if it's permanent or you have a recovery after a few years of not having fire in the...
Sorry, I did not understand clearly. Actually, I don't know a lot of permafrost fate under the fire. I don't know. We try to understand it, how to change and what's the dynamic of the permafrost under the fire during the time after this. How it changes, go to deeper or to recovery and something. It's just to need to be more investigation this way.
Thank you very much.
Nice presentation. Thank you, Katerina. Is it working? Okay. No, I was wondering about the mercury in a liter and a mineral fraction that you chose. The relationship, this one, I think, yes. You see that the slope in the first figures with the liter is higher than the mineral fraction. It means you are accumulated more mercury in the liter fractions than the mineral fractions.
Yes, mercury mostly accumulated in the liter fractions due to the deposition from the atmosphere and due to the capture mercury when plants grows. And after plants die, it's mercury with the organic component go to the down and can be conserved in the mineral horizon. It's not geogenic fluxes from the deeper and so on. It's mercury flux from the atmosphere. It's
Okay.
yes.
Yeah, because I was wondering if you decompose the liter in organic carbon, should be the same rate. But okay, your explanation now explain a little bit more. So in my second question is about the CMIC ratio to soil organic carbon ratio. In the after fire, you yeah, this one.
This
No,
No?
no.
Previous.
The previous one, yes. Yes, I think. It's mean I don't follow very well this slide in the app. Is CMIC divided by soil organic carbon higher than 150?
Oh, of course, it's my mistake. It need to be 1 point in minus something. I
Okay.
don't forget about it, of course.
Okay, this is 100% or close to
Of course.
100, okay? Okay.
Yes, I agree.
I then just follow up on the microbial biomass. You see it's decreasing, it's increasing again. Do you know a little bit which biomass it is? Is it bacteria or fungi? And who recovers better after a wildfire?
Actually, now I can't say what kind of biomass it is. We just measure the total amount without any separate for the fungi and microbes. I don't know what kind of.
Yeah, yeah. And then just a speculation, would you expect, because fungi, yeah, they're pipelines and they transport things, would they be a source of transport of the mercury from the litter layer to lower layers in the soil?
Yes, yes, I agree. It could be, but I did not think about.
Okay.
No, thank you very much.
Thanks.
Last is always best, I guess. This isn't really a question, but it seemed, there's two basic ways that plants deal with mercury. One is, well, if they're resistant, they're either mercury accumulating plants, which then ends up in the litter, or they're methylmercury generating plants, which then puts it back into the air. And it would be a really fun project to figure out, when you have revegetation of the tundra after a fire, if those plants are predominantly one form of mercury generating methylmercury to volatilize it, or if they're accumulating it and then leaving it back into the soil as the litter. But I mean, if you have 35% loss of mercury, it has to go somewhere. The most likely place it goes is into the air, right?
Yes, it's mercury really accumulated by the vegetation, and after the fire, it could go back to the atmosphere. Of course, it should be more clear understanding what is processes and what is not only how change their content, and maybe we should to think how change their pool of mercury.
Yeah, I mean,
Because it's really strong depends of how many vegetations there are.
I mean, you have a rich research environment to look at. It's a wonderful system to look actually at how soil mercury management occurs, because you actually see a profound change. And so the fire, and I don't encourage fire anywhere. I started my undergraduate career working for the fire lab at the U.S. Forest Service, so I don't like fires. It's innate, but nonetheless, fire provides, in this case, a really great research opportunity to understand mercury cycling, which is critically important. So, anyway, I enjoyed your talk. I think it's
Thank you, Ekaterina. We will continue with the next speaker.
Good morning. We are continuing with the presentation of Dr. Roland Bohr. The title, Green Phosphate.
Yeah, thank you all. I'm going to talk about green phosphate or greening phosphate, and I will tell you why. Okay, just a bit for the obvious, that phosphorus is an element, a symbol P, atomic atom number 15. You can never find it as a free element. It has a concentration of about 1 in 1,000, and it's vital to life and it cannot be substituted. And for those who work in agriculture, they know that it's probably after nitrogen the most critical element in agricultural systems. Therefore, you can say that in some ways the world food security depends on phosphate fertilizer. There is also another aspect with phosphate which is different with nitrogen, because nitrogen we can make via the Haber-Bosch, but phosphate we basically mine. So it's a limited resource. So maybe I would like you to contemplate for yourself, where do you think, does your phosphate come from? And how much do we have of it? This is the list of the top 20 suppliers and the lifetime of the supplies. You see some obvious countries, China, United States, Russia, some less obvious places, Morocco, Jordan, Brazil. But basically, when you look on the other side, when you look at the lifetime of the supplies, you see that basically all big countries have a supply less than 50 years. There's only a few countries which have more, and that's mostly Morocco, which has most. But it also must make you think that within the next 50 years, if we don't find more phosphate and more rocks, we have probably several billions of people who are relying on phosphate for their agricultural crops that is not there anymore. Or that they have to get it from specific countries. So, just to make it more simple, if you look at the peace stocks globally, basically you see there's only one country which has a lot of stock, and that's Morocco. All other countries have a little bit, yeah. So basically this is a situation where over time we will see there's only one country basically where we get a pea fertilizer for, and that's Morocco. Of course, there will be some developments in finding new ways of phosphate. This is a little bit of a European problem, but every story has its reflection later on over time in other countries. Yeah. So, basically, the story is that if you look, we always be using more and more of phosphate. It's becoming more and more expensive, and we need it. Europe has a particular problem because it has no phosphate mines. It has a few very small Finnish mines, yeah, but it's really heavily relying on imports. And as I showed, there's only a few countries which basically have phosphate rocks to make phosphate from. So we have geopolitical risks that if there are issues with countries, we may not get supply of phosphate for pea fertilizer. So also phosphate ore is a finite resource, and we're using more and more of it. All of us who work in agriculture know that we cannot just apply phosphate. We can only apply so much under certain conditions. We have to be very, very specific. Yeah. But whereas we very much regulate on one side, it's very unclear where does this rock phosphate come from? How has it been extracted? What is the chemical composition of it? So we have like a very opaque part of this system, of the cycling, which we don't know. I think if I would ask you, where does your phosphate rock come from, which your company or your country uses, very few people would know that. It is really hard to find. It's very, very, very difficult to find out. The best guess would be Morocco. But, for example, when I asked people in the UK who are very much involved in phosphate fertilizers, they didn't know where their phosphate came from, which was produced to make pea fertilizer from. And the same in many countries. Also, it's even more difficult because rocks from different countries are blended in countries, and we don't know. So my talk is a little bit about this other side of the story. It's even worse that if you looked in the Guardian of June 2023, we find that, for example, since, because of the war we have in Europe, there's less supply from Russia, which is then filled up with supplies from Syria, which itself is a really bad situation because, as you know, there's been a lot of war in Syria, internal strife. And so in a certain way, farmers in Europe, without noticing, are contributing financially to the Syrian regime. And this is not acceptable to me. There's also other issues. When you apply phosphate, you also apply the things which come with the rocks. In particular, I would like to make attention in my talk about uranium. Whereas all other elements are regulated, you cannot just apply a phosphate rock. You can only have so much cadmium, so much. Uranium is not, there's no limit on uranium. There's never been any limit on uranium, neither in Europe nor in any other country of the world. In Germany, a proposition was made of about 50 milligrams of U, which has never been discussed in the German parliament. But if you see, you add about 700 tons of uranium to soils in Europe alone, you know. Think about how much that in the world would be. The most dangerous bit with uranium is the leaching into the water supplies, and then through that it gets into the food chain to humans. So basically to say, we have the phosphate mining, you get all these elements like cadmium, chromium, arsenicum, lead, But these are all regulated, Uranium is not. When you mine it, you make pre-fertilizer, you already get an enrichment. Every time you apply mineral pre-fertilizer to land, you apply some uranium. It's a trace element. It can go in the plant, to the plant in the humans, or it can go into the soil, in the drinking water, and get into our bodies. So my wish is that we would see a greener pre-fertilizer. Just like when you go to the shop, you can decide on your coffee. Do you buy the cheapest coffee? Do you buy organic coffee? Do you buy, At the moment we have no idea where the pre comes from. We have no option, we just get what we want, and so. It's even harder now because it's getting more and more expensive, so farmers just take what they have. So I want to make a case to, that we should know more about where the pre-fertilizer comes from. From my point of view with the uranium, I would like it to be less than 50 milligrams of U per kilogram of P2O5. I think the suppliers of phosphate rock should be more open about the damage they do to the environment, the human impacts it has directly or indirectly, before it enters the farm, Why do we at the farm have to work so tightly with our P, if before it's already done dealt with the environment, before it actually gets on that farm? I also make a case, and I will explain later, to replace the mineral fertilizer, mineral P, with organic. And also, like always, this is what it's about, we must recycle, That's why in Europe we see a lot of need to make P into struvite and other recycled P, which we then apply to land. Okay, this is a bit the same as I showed before, so basically you apply the P to the rock, it goes in the soil, sometimes it gets complex, sometimes it goes in the water, sometimes it goes up in the plant, and sometimes you lose it, But you can basically, for every 100 kilos of P, in 100 years. It's not a lot, but we are, we've been at agriculture for quite a lot of time, and we've been adding minerals for a long time. There's some good news. Basically, plants don't need uranium, don't like it, so they don't take it up. So this is not the pathway we should be worried about. There's also more positive news. You can just remove uranium from P-fertilizer. And in the old days, that was done because uranium was a valuable element for all kinds of reasons, for nuclear reactors and things like that. But nowadays, as the uranium price has dropped, we don't do it anymore. But it's still feasible. Some countries still do it. We know, for example, that China is doing this because it's a cheap way of getting uranium. Israel's doing it for other reasons. And so it can be done. And the only question, of course, is when you take uranium out, what do you do with it? There's not that many options. As you say, you can use it for nuclear reactors, or you can use it for depleted uranium in weapons, which is not what we want. And for the rest, there are not that many. There used to be some uranium glass, but we cannot use it that much anyway. So, and also when you take uranium out of phosphate, you immediately fall on the IAEA controls, because any uranium in the world is controlled by the UEA about, so we know where all the uranium in the world is in case of these non-nuclear proliferation issues. So it's not an easy element to recycle. There's also a little bit of good news or bad news that it's not always the same, you know. So some older rocks, they have less uranium than some newer rocks. So the rocks which are less than 300 million years old, they will have less uranium in it. So this is a map of all the sources of phosphate rocks in the world, as we know at that time. So you will see. So this one is the United States, that they have very young rocks for phosphate, so they will have a lot of uranium. These are the African countries and the Middle East. They will have slightly less, but still a lot. And this, for example, is China, which has older rocks and will have less uranium in it. Okay, now I want to just to test this, how effective this was. We did some look at some long-term work to see how much uranium comes with the phosphorus. This is the general level of uranium in phosphate rocks. And you see basically that igneous rocks, they have less uranium, whereas sedimentary rocks have more. You can also see there's a very wide variation, going up to about 250 from really small amounts. So depending on the source, yeah, you can have a lot of variation in uranium that comes with your pea fertilizer. Problem here, of course, is if we have a chance, we would select these mines on this side, but we don't know. Yeah, we just get what we get. Also, why there's a good reason to use more organic, because there's much less uranium in organic materials. As you can see, that whereas the mineral fertilizers have, up to like 100, all the organic sources are below the level of the global level. So it's also good. It's also a little bit of another reason why we should incorporate more organic fertilizers in our agricultural systems. So basically, yeah, you have at least 10 times more uranium when you use mineral fertilizers. This is how we did it. This is how you've got different fractions, but we measured basically the total uranium, and we measured the acid extractable, and that's more the anthropogenic U input. So this is how we look. We used sodium lithium boride for the total, and then a microwave extraction for the acid extractable. So these are some of the experimental sites we looked at. We had one grassland, we had 2 arable lands. These are all in Germany, by the way. And you can see all of these, there really are long-term experiments up to nearly 100 years. They have a variety of treatments. They have with and without pea fertilizer, or they have, combination where there's one treatment which has pea fertilizer and the other one doesn't. This is the Rangen grassland. If we look here, we have on this side the uranium content, this side the treatment. And what you see is the treatments which have P in it have a higher uranium content than the one which don't have P in it. So basically there is an accumulation of uranium coming from the pea fertilizer. When you set that out, you will see that there is about 10.10 milligrams per kilogram more over about, 70 years. So the U concentration in the pea fertilizer increased with about 0.1 milligrams per kilogram after 75 years. Then we looked at Turo, another long-term experiment. Here we also looked at depth. And if you would look at it more, you would expect that with increasing depth, the uranium gets higher. But here, in the top, we have this inversion. And this inversion is very much likely due to the P fertilizer or the other treatments we do. But you see, the one which have P in it, again, have higher uranium than the ones which we don't. Okay. Then we had another long-term experiment, the Askov, which is really one of the longest experiments we had data from, about nearly 100 years. In the top, you see the increase over time of the total uranium. Again, here you see, when we did in 3 periods, that after about 100 years, you have more total uranium in the soil than in 1923. Note that there was some earlier samples, but we did not have access to those. And then here below you see the acid extractable situation. And basically you see again here we have the uranium concentration, here we have over time. And you see that in all of them there is a linear increase. But the increase is most where you have the NPK, where you have the animal manure and you have the control, they move in the same direction, but there's no difference. And that again confirms that we need, we need animal manure or organic pea fertilizers to limit this issue. There is this, to summarize, so basically we found, when we looked also at other data, all other data from we could find in the literature, that grassland accumulated between 7 to 47 micrograms per kilogram per year, arable lands 1 to 18 micrograms per kilogram per year. We found that the ones we looked at, they were all very, very low, so there's no real worry there. Also, when you look at the rate of P apply, you see that, that most of these had about 30, but there were only a few which had higher rates, and you see the higher rates are mostly reflected, leading to higher uranium accumulation, which makes sense. We have some odd ones, I'll come back to you. You can see here Japan's very high. But the basic reason why we did not see this uranium accumulation, and maybe we should have known that before or looked at it, is that, but that's also because we didn't know the source of the fertilizers, because we didn't know what kind of P they used. So basically with Rangan we did know that was basic slag, which is a residue from iron, and that has a low P, a low uranium content. With Turo and Askov we found out by talking a lot that that most likely the P fertilizers they used were from the igneous type, and that they would therefore be very low in uranium. That's why we did not see a lot. But what happened, we did not see a lot, but we could still measure it. Even with these lows, we could see it, so there is accumulation. So we can say that for these situations, if you use these igneous rock with low P, there's no real issue. Then we found a very unusual experiment in Iceland, one of the most northern country in Europe, where they also did some long-term experiment with P fertilizer. and on a rather unusual soil, on an endosoil. And endosoils, they are, very important agricultural in Japan, Iceland, also here in Chile and New Zealand, and some tropical. They're a bit different because they have a high organic content, high water capacity, and lack of cohesion. They have an inherent low P availability, so people tend to apply a lot of P fertilizers there. Yeah. So what here we see how much was applied in these two long-term experiments. And these values about 80, 39. It's on the somewhat higher side. But the critical point is here, look at these bulk densities are very, very low. Yeah. High organic matter. Yeah. There is absolutely in the world no limit on how much uranium you can have in your soil, except one. There's only one country which has specified at which point at so much uranium you would not be able to use your land anymore. And that's Canada. And it's at a limit of about 23 milligrams per kilogram. And interesting what we found here in these soils, yeah, we come to about 15. So we really come very close to this limit, this only limit, yeah. And that, as you see, they stopped these experiments quite early on. If they would have continued to now, we would have reached this limit, yeah. So these soils, the agricultural soils in Iceland, on these endosoils would have not been suitable for agriculture if they apply the Canadian, level. But as I say, there's no country which sticks to this level. So again, here I laid it out a bit differently, how much the annual input is, 130, 149, 319, 310 micrograms per kilogram. So to summarize a little bit here, there were large amounts of P fertilized relatively to maintain the grassland because of low P availability. Because of low bulk density in endosoils, it concentrates U in the upper surface. They have a high organic matter, which promotes the retention of U. On the grassland, there's no tillage, so you also get a concentration in the top. But still, it is a bit of an interesting situation here that I would be very interested to see and get more samples from these endosoils, because I think somebody may suffer from severe U contamination. And I think, we would like to be calling on checks and regulations of these, how much uranium is in these P fertilizers. So a little synthesis. So when we put all the data together, and we looked at the inputs with low and high P, with uranium, so we have two lines. So for the low P, you sit at this rate here. At the high P, you sit at this rate. This is the global average. But you see here, all these endosoils of the world are sitting really above that, yeah? And of course, we also even have a break here, So to summarize, basically, this is the global increase. It's 0.25 micrograms of U per kilogram of P per kilogram per year of what's applied. And we finally looked at the broad bulk, which is the longest experiment. And here again, we saw the same situation. If you apply mineral P fertilizer, you get this increase. Yeah. So this is not a problem which is solved. It's a problem which is continuing. So now we have the issue. We know what it is, but now we have to do something about it. Yeah. So and how to do that? That's very difficult because there's no political will or there's no mechanisms or so. And they try to reduce the cadmium, but the uranium has never been dealt with to reduce it in P fertilizer, and it's unlikely to be done so. Okay. I see a yellow light, so I will briefly. So basically, U concentration in P fertilizer is the primary effect of U accumulation in soils. It's of course affected by management, by fertilizer rate, more fertilizer, more in, land use, and the soils itself. I think these factors should be considered, regulated at political level to limit the environmental risks by free fertilization and induced contamination. I think also it's something we cannot solve, we need to do together, and I think, yeah, you know, the P for doing this together. Okay, and this was not done by myself. Most of the work was done by one of my PhD students, Yatje San, with a lot of help from other people. And yeah, I did it a little bit myself. And there's probably a lot of other people who are missing here. So thank you all for listening. And yeah, if you have any questions, I look forward to having you.
Thank you for your nice presentation. We have time only for two questions.
Thanks, Roland. Thank you very much for your very nice talk, very interesting. I was wondering about the speciation of the uranium in the soils.
Yeah.
And you also said that plants would not take up the uranium too much. So I was wondering why it's forbidden to do agriculture when you have a certain uranium level exceeding.
Yeah, that's why probably the level's set quite high. But I think the danger with uranium is not the plant, but it gets into the drinking water. You know, I think nowadays most of us will get 70% of our daily uranium supply to our drinking water already. So.
And the speciation is this
speciation is
organic?
No, they have a uranium-2 and uranium-4, and one of them is quite stable, and one is more mobile. So it depends also on the pH and some soil conditions. Yeah, but I didn't want to go into that much detail about that.
It's occurring in inorganic forms only, or are there also organic forms?
There will also be some organic forms, but I think there's not that much. Maybe some knowledge, but not that much I'm known about, you know. So some of this work on uranium is not very public, let's say it this way. Yeah.
Roland, thank you very much for this overview. For the long-term experiment in Askov, Denmark, you clearly show that also without phosphorus application, uranium increase, and the rate of the increase is nearly the same as with phosphate fertilizers.
Precisely.
How you explain this?
We have two explanations. One explanation is that there is uranium coming into the atmosphere. We try to measure that, but that's really not the case. So we think that this is got to do with that occasionally all soils are limed in Askov every 5 or 10 years, and there's a little bit of uranium in there, and that's why we see this increase. But we also had a bit of trouble in the beginning explaining that, you know. And maybe there's also something which comes into the atmosphere, but you know, we don't measure it, or nobody measures it, you know, so yeah. But thanks for spotting that, and I wanted to comment on that, but yeah, now it's asked question is, thank you.
Thank you, Roland.
Okay.
The next speaker is Eulogio Castro from the University of Jaén in Spain. Thank you for coming, Eulogio.
Thank you very much. I would like to thank first the organization, the doctorate program, and the University of La Frontera as a whole for inviting me here. It is an excellent workshop with an excellent organization, and I am very happy to be here. My talk today will deal with biorefineries, and particularly the use of residual biomass as a feedstock. So I would like to start with the definition of biorefinery. There are a lot of them, but one of them is a facility that integrates biomass conversion processes and equipment to produce value-added fuels, electricity, and chemicals. Excuse me. So, most of you are also aware that there is a parallelism between the conventional oil refinery and the biorefinery. So the oil refinery can produce a wide number of products, including energy, chemicals, materials, from fossil fuels or from fossil materials, while the biorefinery can do the same starting from renewable materials. And that's the key point here. I would like to show you also this figure that was introduced by Novozymes, and it is an estimation of how much agricultural residues we will have available in 2030, divided into 8 main regions in the world. So you can see here, for example, that we have more than 150 million tons in Europe, more than 200 million tons in South America, just of agricultural residues that must be adequately handled every year. So, globally, Novozymes thought that more than 900 million tons of residues will be available every year. That means that half the gasoline needs of the world could be covered if we were able to convert all these residues into biofuels, for example. So, let me introduce myself. I am Eulogio Castro from the University of Jaén in Spain. Spain is located in Jaén is located in South Spain and in the region of Andalusia. So, what we do at the University of Jaén, and this is the focus of my talk today, is dealing with agricultural residues. As you will see, we are focused especially on the residues from olive tree cultivation and olive oil production process, but we have also worked on several other agricultural residues. For example, rapeseed, for example, eucalyptus, this is almond tree, this is sorghum, this is brewery spent grains, and so on. So what we do is try to get full advantage of the sugar content of residues, of agricultural residues. You see, you know that more or less average cellulose, hemicellulose, and lignin account for nearly 80% of the plant material, and sugars are located in the plant cell in forming cellulose, hemicellulose, and also there is another polymer, lignin. So we are dealing with all these 3 parts of the materials. Let me show you something about the region where I come from. This is a picture, a Google Earth picture above virtually any point in my province, in Jaén. So if you see here, there are a lot of little points. Coming down a little, you will see that every little green point there is an olive tree. So we usually say that we have 70 millions of biorefineries in the province of Jaén, each one of these points. So this is the usual landscape. Looking at any direction in Jaén, we have more than 600,000 hectares of olive trees, 2.4 million in Spain. Olive trees is not exclusive from Spain or from the Mediterranean countries. Nowadays, there are more than 12 million hectares around the world in many countries. And If you are not very familiar with the process of olive oil production, let me explain a little how it works. So we have olive trees, we do the harvesting. After the harvesting, we do the pruning every year. Well, actually every 2 years on the same tree. So pruning produces a huge amount of biomass, approximately 2 tons per hectare. Well, so we do harvesting and we pick up olives together with leaves and branches, and we go to the olive mill. In the olive mill, the first operation is cleaning, and we separate leaves. Sometimes, if we need to wash olives, we get also wastewater that is directed to an aerial pond to evaporate slowly. And clean olives enter the process. The process mainly includes milling, mixing, and centrifugation. The typical centrifuge is called here the decanter. And the decanters do the separation between oil and the rest of everything, what we called olive pomace. So oil is then submitted to a second, to a washing with water in a vertical centrifuge, and we get the olive oil. This washing water is also sent to the aerial pond, while the olive pomace still retains 2 to 3% of residual oil that deserves to be extracted, but now we must use a chemical process for that, using, for example, hexane. Before that, we need to recover the pits and dry, do the solvent extraction, distillation, and we get the raw pomace olive oil, which is also commercialized after several steps of refining. And after solvent recovery, we get the last residue, which is called the fatted olive pomace or orujillo. So this is olive pomace, this is the pits, orujillo, water, pruning, leaves. We have several different kinds of residual biomasses, and we do research on every one of these biomasses. So at the end, we have pruning, and we get biomass from pruning. This is obviously located in the fields, and we have then olive leaves, olive pits or stones, olive pomace, the fatted olive pomace, and olive meal water. And this is what we are researching on, because we think it is possible to establish a biorefinery based on each of these biomasses to produce a wide range of products. For example, bioethanol, of course, that was our first target, oligosaccharides, antioxidants, xylitol, succinic acid, ceramic materials, and so on. And this is what we called the biorefinery based on olive-derived biomass. So, how much of these materials are available for everything? You can see here, based on the production of olive oil in Spain, for example, in average values, we have 3 million tons of pruning biomass every year, 1 million tons of olive stones or pits coming both from olive oil production process, but also from table olive factories that are quite important also in Spain. 1 million tons of leaves, 5 million tons of olive pomace, 2 million tons of orujillo, or pomace oil, or pomace dry residues, and an indeterminate amount of wastewater. So, in the first new slides, I would like to present to you what we are doing with each of them. Starting from biomass, from Pranin, this is the schematic of the conversion process based on a biochemical approach. There is also a thermochemical approach, as you know, but we are particularly working on the biochemical approach. So we have these 4 relevant steps: pretreatment, enzymatic hydrolysis, conversion, fermentation, usually fermentation, and then separation and purification to get the bio-based products. So we started 15 or 20 years ago with the biomass from Pranin, because that represented a serious environmental problem, because Pranin is generated only during 1 month and must be quickly eliminated to prevent vegetal diseases. And the usual way of doing that is just burning in the fields. So we decided to see if there was some alternative. And we applied the conversion scheme. So we did a pretreatment on the biomass. We got the pretreated solid that was enriched in cellulose. We did the enzymatic hydrolysis to release glucose and then fermentation to produce ethanol. 15 or 20 years ago, bioethanol was a very interesting product. It is still now, but perhaps not that much. The thing is that that was our first approach, but we realized that there were the liquid fraction issued from pretreatment with interesting compounds inside also. So we look inside the liquid fraction containing also glucose and xylose, but we detected that there were compounds exhibiting antioxidant activity. There were also, depending on the pretreatment, sugars in form of oligosaccharides that were also very interesting for several applications. And there were also C5 and C6 sugars that could be also directed to fermentation with some particularities, okay? And in addition, there was the lignin-rich residue that could be also used for energy or for chemical production. So that was, you see here, a summary of our first proposal of a biorefinery based on pruning biomass from olive trees. Olive tree pruning biomass was extracted with water, and in that fraction we could already identify antioxidants and sugars, of course. Then we did the pretreatment. The liquid fraction contained as well oligosaccharides and antioxidants, and the pretreated olive tree pruning could be directed, for example, using a simultaneous saccharification and fermentation scheme to produce ethanol. And we can also prepare some particular materials, as I will talk later. So here you can find the mass balance for this first approach on biorefinery based on pruning. So ethanol, 220 kilograms per ton, if we were able to convert all sugars, those coming from the cellulosic fraction and from the hemicellulosic fraction. This is not easy, but it could be. 100 kilograms of oligosaccharides depending, as I mentioned before, on the pretreatment, particularly if we do not use acid in the pretreatment, just water. Antioxidants, 18 kilograms, and still we have a lignin-rich residue of 150 kilograms. That was with pruning, biomass from pruning, but in the olive mill, we had already accumulated the olive leaves that were separated from olives. So we decided to study a little more in depth also this fraction. And as you can see here, the main feature of olive leaves is the extractive fraction. That was also a challenge because this fraction was almost completely lost when submitted to the pretreatment. But anyway, I will not come into more details, but we did also a proposal of what we could do with olive leaves based on this general scheme. And for example, one of the approaches led us to use a steam explosion pretreatment, and the products obtained were natural antioxidants, among them hydroxytyrosol, that it is very interesting, oleoprotein as well, oligosaccharides, fermentable sugars, and still we had a pretreated solids of about 50% of the initial weight of olive leaves. Olive pits are also recovered in olive mills, and this is a product very appreciated because it is used as a biofuel. Until some months or perhaps years ago, there were a ship every week departing from Malaga port to London because they use olive pits there to produce electricity. And that was, there was a compensation because of the renewable oil. And you can see here the main composition of olive pits that represent 10 to 15% of the weight of olives. And as you can see here, xylose is one of the compounds more represented in the composition of pits, sometimes near 30%. So we think that before using olive pits just to produce heat or electricity, we could see if something else could be done with that. We have several papers on that, but for example, here you have one of the options. It's an option including several steps before pretreatment, trying to get full advantage of, for example, the antioxidant properties of water extracted liquid, and then separating a pentose-rich liquor before doing the pretreatment itself directed to the cellulose fraction. But anyway, there are also a number of products that could be obtained from olive pits. More recently, we have also tried to widen the range of products that we could obtain from olive pits. And for example, here you can see the mass balance of a process for producing fulgrar, a platform chemical also very appreciated. And this is also an option using acid hydrolysis and then microwave treatment. And for example, here you can see that 9 grams per 100 100 grams of olive stones could be obtained. So that was the general scheme. I have talked a little about biomass from pruning, olive leaves, olive stones. Let me now comment a little on the other residues. Extracted olive pomace. This is the last point of the process. This is the last residue we obtain in the facility where pomace olive oil is extracted. That's also used as a biofuel, but we decided also to explore all the possibilities of using with some other objectives. For example, here we saw that ethanol can also be produced from the hemicellulosic fraction of that, and that was also a challenge because you cannot use, for example, Saccharomyces for producing ethanol from xylose unless it is genetically modified. So we use here modified Escherichia coli, E. coli, to produce ethanol. And at the same time, there was also a valorization for antioxidant compounds in the same scheme. The lignin was also one of our objectives here because lignin was the last product we considered in our research scheme, but now we are dealing with that also. And so we propose a biorefinery to obtain antioxidants, lignin, and sugars from this exhausted olive pomace. You can have more information if you are interested here in the references. Again, the process, I will talk now about olive meal wastewater. There are 2 points where wastewater is generated in an olive oil facility. First one, this is the general scheme, again, of producing olive oil. Olives are cleaning. Here, leaves are eliminated. Sometimes olives are washed, and then we produce the washing wastewater. And then olives are grinding, mixing, and separating by centrifugation in the decanter. And we get the 2 fractions, the oil and the rest. That means the pomace. So the oil was directed to this vertical centrifuge and was washed again to eliminate the rest of skin and pulp and so on. We have our main products here, the extra virgin olive oil, and we get also the olive oil washing water. So the wastewater, both are usually sent to an aerial pond like this. And they wait there for several months to evaporate. So what we decided to explore here was because Jaén, in addition to produce a lot of olive oil, produce also ceramic materials. It's the second location in Spain producing bricks and ceramic materials in general. So this is the general scheme of producing bricks. It is a simple thing, let's say, just using clay with water, forming the brick, and then eliminating water. So this is the olive oil extraction process again. Olives, washing, crushing, mixing, decanter, oil washing, olive. So we have here washing water olive cake or olive pomade and olive oil washing water. I think you have already imagined what we did. Substituting tap water using here by washing water, olive oil washing water, or even olive cake. And that was very good. We did that in collaboration with one of the main ceramic industries in our region, and we arrived to 3 different products. When we use olive oil washing water, we produce a brick very similar to the conventional one, but the ceramic industry needed less heat in the kiln. So there was a fuel savings in the kiln. That was very good, of course. If we use olive pomades instead of a reduced amount of clay, then we got a different brick, not the same brick, a brick with special properties, low density, acoustic and thermal, insulation properties, and we also get fuel savings in the kiln. And for example, we use also ash, the ash from olive tree biomass combustion. And here it turned out that the best thing was not the brick itself, but the source of potassium. And this allowed us to fill the Spanish patent on the recovery of potassium from these ashes. So, in addition to that, we did the complete Aspen Plus simulation of the brick factory, and the good thing is that the brick company could get a reduction in CO2 emission. For example, using olive oil washed water, 2.3% of reduction in CO2 emissions. But if we use olive pomade at 3%, only 3%, the reduction was 6.5%, and 13% if the olive pomade was 6%. This is very important now, especially in Europe, because emitting CO2 is very, very expensive there. So that was one of the results. So, in conclusion, I have entitled this talk Research on Residual Biomass, and I would like just to summarize the some of the bioproducts we can get, and this is some of the starting material we have been dealing with. And as I think I have a couple of minutes, I would like just to introduce our last research project. This is based on wine residues. So you probably know that Spain is also a big wine producer. And Jaén is located here, and here, this is La Mancha, the biggest wine-producing region in the world. So, we have now a project in which we are going to study what to do, particularly with vine shoots. There are like 3 tons of vine shoots per hectare, in addition to other products or residues, and this is the average composition, and we are trying to do the same using the biorefinery schemes we have already worked on to fractionate and to get a number of products from all of these fractions. This is a project in collaboration with two other research institutions in Spain, CIEMAT, CSIC, and the University of Jaén, and I hope you will have news from our side in terms of publication or something. So thank you very much. This is my research group. Some of them are fortunately not anymore with us. That means that they are working somewhere else. But I would like to thank all of them, present and past members, and all of you for your attention. Thank you very much.
Thank you, Eulogio. We have time for 2 questions from the people. Here.
Thank you for your presentation. Very interesting for me. It's rather new. I was wondering if you try life cycle assessment in order to see if you put more energy to produce these products compared with the benefit that you get from those products.
Yes, we do. The thing is that I am using an old version of this general scheme. Let me see, here. In the new one, I have also here the life cycle assessment. But we do the techno-economic evaluation as well. I didn't have much time to talk about that, but the life cycle assessment is always a part of any of our projects. Actually, if you ask for some money for researching, you must include life cycle assessment as a must, and the techno-economic evaluation as well. So we are doing that also, yes.
There are other questions?
Just to have a general question, are there a lot of pesticides? Are there a lot of pesticides used in olive cultivation?
In relative terms, no. Or in relative terms, yes. It depends on. There are some of them, of course. There are some of them, but you know, this is. I would like to say that this is a very controlled sector, even because you know. A great part of the olive oil production is exported to many other countries, and in the European Union, the controls are really strict. Yeah.
And does this affect somehow your processes?
No, not really, because our processes start almost always with 200 degrees. In the pretreatment. So I think that even if we use microorganism, but it is in a later stage. So I don't think there is an influence of any pesticides on this.
Okay, thank you.
Okay.
Thank you, Eulogio.
Okay, thank you very much. It's time for our presentation. Next speakers. Cristina Area, de la Universidad de Misiones, Argentina. Nos va a presentar su trabajo titulado 2G Biorefinery.
Good morning, buenos días. Voy a hacer la presentación en español. I will speak in Spanish because I want some people to understand me. Bueno, en principio, muchísimas gracias, Amara, por la invitación y a la organización del evento, que está fantástico. Realmente la idea de hacer este evento para estudiantes está buenísima. Así que, bueno, un poco mi presentación está dirigida a los estudiantes, ¿sí? Creo que, bueno, mucho de lo que voy a decir ya lo dijo Eulogio, pero lo voy a decir en castellano. Así que bueno, yo me presento, soy la doctora María Cristina Area, soy investigadora principal del CONICET, que es el Consejo Nacional de Investigaciones Científicas y Técnicas de la Argentina, y soy profesora titular de la Universidad Nacional de Misiones. Mi lugar de trabajo es el Instituto de Materiales de Misiones, del cual soy directora, y mi lugar específico, mi grupo, es el programa de celulosa y papel. Yo soy doctora en ingeniería papelera, máster en ciencias de pulpa y papel, entonces voy a explicar un poquito por qué una papelera se dedicó a las biorefinerías, ¿sí? Bien, entonces voy a hablar primero un poco de bioeconomía y biorefinerías, la biorefinería forestal, las actividades nuestras del programa de celulosa y papel y nuestras investigaciones. Después voy a hablar de un gran proyecto que tenemos que se llama Biona y las conclusiones. Bien, yo creo que todos saben a qué se refiere el término bioeconomía, ¿sí? Básicamente tratar de cambiar todo lo que es base fósil a base renovable. ¿Sí? Entonces, en esos términos surge la biorefinería. Y la biorefinería es una estructura que integra procesos de producción, equipamiento, y la idea es producir bioproductos como biopolímeros o químicos de plataforma, biomateriales como plásticos, textiles, materiales compuestos, y biocombustibles como etanol y biogás. O sea que es un concepto análogo al de las biorefinerías de petróleo, pero basados en fuentes renovables. La biorefinería forestal, en particular, comienza con una producción forestal sostenible y toma ventaja, o sea que aprovecha todo el árbol, no solamente el tronco, como la industria tradicional, sino también los residuos. Y genera productos tradicionales, como puede ser papel, tableros, ¿sí? Y un rango de productos de alto valor agregado, o sea que valoriza la materia prima forestal. Entonces, cuando nosotros hablamos de lo que es la cadena de valor forestal, arrancamos con lo que sería la biofábrica o el lugar donde se producen los plantines, después tenemos los viveros, la plantación, con los troncos, siempre, digamos, la producción tradicional, que son la madera cerrada, la madera debobinada, también tenemos los tableros con los residuos, también la energía de biomasa, y con las fibras, pulpa, papel, cartón y tableros de fibra. Ahora, a todo este esquema tradicional se agrega la producción de bioproductos, biomateriales y bioenergía con los residuos. ¿Cuál es la importancia que tiene esto? ¿Sí? Básicamente en cuanto a la economía y al medio ambiente. Bueno, si nosotros vemos la pirámide de valor de estos materiales, nos encontramos con que la base está formada por los desechos, o sea, hay mucho mayor cantidad de residuos producidos que de lo que es la materia prima tal cual. Entonces, esos residuos no tienen ningún valor, muy bajo valor, y se producen en grandes cantidades. De hecho, por ejemplo, para comentarles un ejemplo particular, en Argentina se producen muchos muebles de algarrobo, que es una madera dura. El mueble de algarrobo aprovecha el 30% del árbol y todo el resto es residuo. Entonces, ¿qué es lo que sucede? Hay residuos, hay mucha proliferación actualmente de lo que es energía de biomasa, entonces hay mucho residuo que se puede quemar, pero ¿qué es lo que pasa? Que los aserraderos están dispersos. Entonces, los residuos no están, digamos, concentrados y el costo de transporte para un producto que tiene valor prácticamente cero encarece mucho el uso. Entonces, es como que no tiene sentido, ¿no? Si nosotros vemos, la bioenergía está tercera, o sea que en realidad tiene bajo valor. ¿Sí? Lo peor que uno puede hacer con el material es quemarlo. ¿Sí? Es preferible producir biomateriales y bioproductos que producidos en poca cantidad tienen un mucho mayor precio. Entonces así surgen los tipos de biorefinería que se aplican específicamente a estos materiales lignocelulósicos, como le llamamos, que son básicamente de lo que llamamos dos plataformas. La plataforma termoquímica, que puede ser combustión, pirólisis o gasificación para producir, ¿no?, nuestros materiales químicos y combustibles, o la plataforma bioquímica, que puede tener dos vías, como los procesos de pulpado tradicionales o los procesos de fraccionamiento del material para llegar a los mismos productos. Si hablamos de la conversión termoquímica, ¿sí?, tenemos la transformación de biomasa en gas de síntesis o en otros compuestos, es lo que llamamos syngas, y se pueden producir, digamos, productos a partir de esos gases, ¿no?, ya sea pirólisis o gasificación. Si nosotros lo vemos acá, nos encontramos con que nosotros podemos producir el syngas, pero el syngas es una mezcla de productos que para llegar a los productos finales, por ejemplo, veamos el etanol, ¿sí?, o cualquier químico, tenemos que pasar por procesos catalíticos muy complejos porque digamos, los compuestos están todos mezclados en el gas de síntesis. Entonces, para llegar a los procesos últimos tenemos un procesamiento muy complicado y sobre todo la separación y purificación de los productos finales es muy compleja. Entonces, la plataforma bioquímica o química es totalmente diferente. Es mucho más complicada. ¿Por qué? Porque tenemos que separar las cosas, no es simplemente quemarlas, ¿no? Pero tiene la posibilidad de obtener en forma mucho más sencilla productos de alto valor. Perdón. Entonces, acá se plantea, bueno, ¿por qué? ¿Por qué producir productos a partir de biomasa compleja si nosotros podemos producir, por ejemplo, etanol a partir del almidón, del maíz o del azúcar, de la caña de azúcar, por ejemplo, que serían lo que llamamos biorefinerías de primera generación? Las biorefinerías de primera generación, o 1G, son las que aprovechan directamente los almidones, ¿sí? Entonces, yo puedo tener perfectamente, a partir del maíz, etanol, un bioetanol perfectamente. De hecho, en Argentina hay producción de bioetanol a partir de maíz y en Brasil hay mucho a partir de maíz y a partir de azúcar, ¿sí? Ahora, ¿qué pasa? Que esto compite con alimentos, ¿sí? Ayer o antes de ayer estaban diciendo justamente que el maíz y el arroz son las dos principales fuentes de alimentos del mundo. Entonces, nosotros estamos usando esa fuente para producir un combustible. Eso no puede ser. Entonces, la biorefinería de segunda generación es más complicada, cuesta más, pero por lo menos tiene la ventaja de que no compite con alimentos y además tiene la ventaja ambiental de que aprovecha residuos que de otra manera muchas veces son quemados al aire libre o son depositados directamente en landfill, ¿no?, en rellenos sanitarios. Entonces, esto que ya había mostrado Eulogio es la complejidad que tiene el material que tiene, por ejemplo, un árbol, o puede tener un tallo de maíz o de trigo o cualquier residuo agrícola o forestal, ¿no? Nosotros, este material está compuesto por fibras y cada una de estas fibras tiene paredes que están formadas por algunos aglomerados de diferentes compuestos químicos que a medida que uno va adentrando, disminuyendo, digamos, o aumentando el aumento del microscopio con el que va mirando, se encuentra con las macrofibrilas, microfibrilas y fibrilas elementales. Las macrofibrilas, fíjense que tienen una composición química en la cual vamos a tener como haces de celulosa y hemicelulosa rodeados por lignina. Si nos vamos a lo que es la microfibrila, celulosa, hemicelulosa, y la celulosa sería la fibrila elemental. Entonces, si nosotros queremos, por ejemplo, obtener una fracción pura de celulosa, nosotros tenemos que eliminar todos los materiales para llegar acá. Eso es, por ejemplo, los cristales de celulosa, ¿sí? La celulosa nanocristalina, en la cual se saca todo. En la nanocelulosa no. En la nanocelulosa llegamos a tener la microfibrila o partes de la microfibrila, donde tenemos algo de hemicelulosas. Y la ligno-nanocelulosa le deja todavía un poquito de lignina. Bien, entonces aquí es donde empezamos a hablar de los procesos de fraccionamiento, ¿sí? ¿Qué es lo que tenemos que hacer para llegar a obtener esos compuestos? Tenemos que fraccionar. Entonces tenemos procesos tradicionales como el proceso Kraft, sulfito, soda antraquinona, que son los procesos papeleros. Y tenemos los procesos verdes de química verde, que son los más nuevos, entre los que están los hidrotérmicos, solventes, fluidos supercríticos, líquidos iónicos y otros, que es lo que va a tratar Fernando en la próxima charla. Entonces, ¿qué es lo que hacemos con el fraccionamiento? Separar el material en sus fracciones. Entonces vamos a tener básicamente una fracción sólida, que en general siempre está compuesta por celulosa, con alguna impureza de hemicelulosa y lignina, y una fracción líquida en la que vamos a sacar, dependiendo el tipo de fluido con el que estemos trabajando, hemicelulosas o hemicelulosas y lignina. Entonces, hay que tomar separadamente esas fracciones para obtener los diferentes productos que queremos obtener. Bueno, y acá está un poco lo que les contaba al principio, cuál es el motivo por el cual los papeleros nos dedicamos a las biorefinerías. Bueno, para contarles un poco la anécdota, más o menos en el momento que yo estaba haciendo el doctorado en Canadá, en la Universidad de Quebec, empezó la crisis del uso del papel por el tema de la proliferación de los medios informáticos. Entonces empezó a vender menos papel y las empresas empezaron a ver qué podían hacer para diversificar su producción y entonces poder empezar a compensar esas pérdidas. Además de eso, o sea, sumado a lo anterior, empezaron a ajustar las cuestiones ambientales. Entonces ya no era cuestión de gastar tanto en tratamiento de efluentes, sino tratar de trabajar en el proceso, extrayendo lo que se podía para que no fuera tanta materia orgánica a tratarse al efluente, ¿no? Y entonces por eso los papeleros nos hicimos especialistas en biorefinería. De hecho, mi tesis doctoral de hace muchos, muchos años trata sobre obtención de floculantes a partir de lignina. Así que ya venía eso desde hace mucho tiempo. Entonces, bueno, de esta biomasa, como dijimos, podemos obtener todos estos compuestos, ¿sí? Que están en diferentes proporciones, a partir de más o menos un 50% de celulosa, 25% de lignina, 25% de hemicelulosa, para hablar así a grandes rasgos. Cada tipo de material tiene diferentes proporciones. Un contenido determinado de extractivos que van a ser de diferente calidad, si estamos hablando de coníferas, de latifoliadas, un pino tiene resina, en polifenoles en el caso de eucaliptus, y materia orgánica que dependiendo de la biomasa también puede ser interesante. Por ejemplo, nosotros hemos hecho trabajos con cascarilla de arroz, y la cascarilla de arroz tiene mucha sílice, que es de muy buena calidad, que puede ser utilizada en circuitos electrónicos. Entonces, cada uno de ellos se puede usar en la industria textil, farmacéutica, química, etcétera, etcétera. Entonces, ¿cuáles son las oportunidades que nos brindan las refinerías? Bueno, en principio, oportunidades económicas, porque vamos a tener nuevos productos con mercados potenciales, son atractivas para inversores, tienen menores costos de producción, ¿sí? Eventualmente, que los derivados del petróleo, menos toxicidad en productos de cuidado personal o alimentarios, usan residuos, por lo tanto tienen ventajas ambientales, reducen el impacto de la materia prima en el ciclo de los bioproductos, se pueden diseñar los productos para ser reciclables y biodegradables, pero sobre todo tienen ventajas sociales. Y esto tiene que ver con que al ser de menor escala se pueden instalar, por ejemplo, en ambientes rurales o cerca de donde se produce, esto que yo les contaba, donde tengo 2 o 3 aserraderos que tienen baja producción, entonces yo puedo instalar una biorefinería para determinado producto que sea apto para esa materia prima, y esa biorefinería puede ser o de los propios productores o los que tienen los aserraderos, o tercerizado, pero aprovechando y dándole valor. O sea que ahí ya estas pequeñas empresas no tienen que regalar los residuos, sino que realmente pueden obtener algo de valor con eso. Bueno, un ejemplo típico de biorefinería papelera lo tenemos en Finlandia, ¿sí? De Metsä Fibre, en Äänekoski, donde esta empresa lo que ha hecho es, a partir de la empresa papelera, de la fábrica de pulpa celulósica y papel, tiene diferentes empresas a las cuales les vende o energía o les da efluentes para extraer algún producto, por ejemplo, y entonces así se va formando una cadena, lo que se llama un ecosistema industrial alrededor de lo que sería una biorefinería. Bien, entonces les cuento un poco de lo que hacemos nosotros, ¿sí? Como les decía, nosotros somos el programa de celulosa y papel del Instituto de Materiales de Misiones. Dictamos cursos de doctorado, una maestría en Ciencia y Tecnología de los Materiales Fibrosos, cursos de pulpa y papel en Ingeniería Química y dictamos una tecnicatura universitaria en celulosa y papel. Tenemos mucha relación con la industria, trabajamos mucho con la industria haciendo servicios técnicos, haciendo trabajos de investigación y desarrollo, pero también desde el 2014 tenemos un convenio con la Asociación de Fabricantes de Celulosa y Papel y dictamos una capacitación a distancia con diferentes cursos, y siempre desde ese año estamos teniendo muchos alumnos de empresas. En investigación trabajamos básicamente en biorefinería, en análisis o estudio de materias primas, pulpado y blanqueo con tecnologías limpias, química de la madera, compuestos madera-plástico, reciclado del papel y productos de pulpa moldeada. ¿Cuáles son nuestras investigaciones en biorefinerías? Hemos desarrollado procesos y optimización de procesos a la soda, hidrotérmicos, con solventes, con fluidos supercríticos y microemulsiones. En cuanto a biomateriales, hemos hecho pulpas de disolución, materiales compuestos, celulosa micro y nanofibrilada, resina fenolformaldehído y bioplásticos como ácido biopoliláctico o biopolietileno. Y en cuanto a bioproductos, xilitol, vainillina, ácidos orgánicos como lebulínico, láctico, succínico y otros, adhesivos, bioetanol y sólidos carbonosos a partir de lignina como grafeno, por ejemplo. Y todo esto siempre haciendo los análisis técnico-económicos, análisis de riesgos, cambios de escala, etcétera, y también algo de análisis de ciclo de vida. Bueno, algunos ejemplos, por ejemplo, en este caso hicimos producción de xilitol a partir de líquidos de tratamiento hidrotérmico, pero lo interesante de este trabajo es que en general la mayoría de los trabajos que uno ve en la bibliografía lo que hacen es presentar la obtención de un producto. Entonces, bueno, sí, todos sabemos que se puede obtener xilitol a partir de silosa. Ahora, ¿cómo la recuperamos y con qué rendimiento? Entonces, el tema es desarrollar técnicas para la recuperación para saber, bueno, a ver eso cuánto va a costar realmente si yo quiero aplicarlo, si yo quiero obtener ese producto. Entonces, esto un poco tiene que ver con eso, ¿no? Con detectar cuáles son los inhibidores y irlos anulando a partir del proceso hasta obtener el xilitol. Con respecto a nanocelulosa, hemos hecho muchos trabajos también en colaboración. En este caso trabajamos mucho con Lerlepamap de la Universidad de Girona, hemos hecho trabajos también con BTT de Finlandia, y este trabajo en particular, bueno, que está acá, es uno de los trabajos del que saqué las figuritas, ¿no? Como les decía, fíjense allá, la nanocelulosa y la ligno-nanocelulosa que tienen un color diferente porque tiene lignina, ¿no? Entonces, diferentes procesos para obtención. Con BTT de Finlandia se hizo un trabajo de enzimático alta consistencia, o sea, un proceso totalmente diferente de los procesos químicos. También hicimos con un convenio que hicimos con la Asociación de Fabricantes de Celulosa y Papel, tomamos empastes papeleros de 5 fábricas y lo que hicimos fue, trabajando de la misma forma, con los mismos aditivos y de la misma forma que las fábricas, agregar nanocelulosa para ver cómo reaccionaban los diferentes tipos de papeles. Y ese trabajo muy interesante porque encontramos, por ejemplo, que en los papeles reciclados, en los cartones reciclados, se puede disminuir el tratamiento mecánico que se hace a las fibras, con lo cual se baja el consumo de energía, o se puede mejorar mucho la calidad del producto final. Este trabajo lo hicimos con PFI de Noruega, que es aplicaciones de impresiones 3D, en las cuales se analizó también la citotoxicidad de los productos para tratar de hacer apósitos de liberación prolongada de medicamentos. El último que está ahí son sensores de humedad, y ahora estamos trabajando en films para packaging, para reemplazar los plásticos de base fósil. Bueno, esto sería un ejemplo de análisis técnico-económico. En este caso, por ejemplo, se trabajó fabricando, en general los trabajos que hacemos es evaluar cuáles son las mejores opciones, las opciones económicas y técnicamente mejores dentro de todos los productos que producimos para que sean rentables. Como les decía, estamos trabajando en bioplásticos para packaging, viendo la degradabilidad, por ejemplo, y uno de los trabajos que encaramos es justamente el tema de producción de biopolietileno 2G. En este caso la idea es tratar de fabricar el biopolietileno que va a ser no biodegradable, pero de base renovable. Entonces, para reemplazar a la base fósil. Hasta ahora hemos llegado al bioetileno, ¿sí? Hemos producido bioetileno en el laboratorio y la idea es posteriormente polimerizarlo. Este es el trabajo que les decía de la producción del bioetileno. O sea que la idea de nuestra producción de bioetanol no es hacer combustible, sino hacer bioplásticos. También pulpas de disolución, como les decía, con lo que hicimos, ¿cómo se llama? Perlas de celulosa y films. Y este trabajo es justamente uno que hicimos con el grupo de Mara, con la Universidad de la Frontera, de Mara y Gustavo Ciudad, en el cual usamos licores de tratamiento hidrotérmico de bagazo, para fabricar, la idea era fabricar polihidroxialcanoatos y terminamos fabricando ácido leulínico. Fue una sorpresa. Sí. Bien, un poco de la infraestructura que tenemos en el programa. Tenemos un laboratorio de microscopía, un laboratorio de química preparativa, un laboratorio de química instrumental, un laboratorio de tratamientos enzimáticos, el de nanotecnología, el laboratorio de procesos y el laboratorio de ensayos físicos. Este es nuestro grupo de trabajo, ¿sí? Somos unos 22, 23. Le estaba diciendo a Mara que somos la mayoría mujeres, como pueden ver. Tenemos investigadores estudiantes postdoctorales, estudiantes de doctorado y estudiantes de grado de ingeniería química que están haciendo sus trabajos con nosotros. Bien, y por último les quiero contar un poquito el proyecto Biona. El proyecto Biona se llama Biorefinerías del Norte Argentino. ¿Y cómo surge este proyecto? Bueno, resulta que nosotros hicimos muchos trabajos en laboratorio, llegamos a los productos finales, los podemos separar todo, pero para poder transferirlos a la industria necesitamos pasar por la escala piloto. Entonces, hace años, hace como 10 años que ando visitando empresas, hablando con uno, hablando con otro, hablando con... Bueno, la cuestión es que finalmente logré ser escuchada gracias al gobierno nacional que por suerte se está ocupando de lo que es la ciencia y la tecnología y entonces generaron un programa que se llama CITES, que son Centros Interinstitucionales Estratégicos. Entonces, si uno se junta entre varios, logra obtener un subsidio de una cantidad suficiente como para poder comprar algo, ¿no? Entonces, lo que hicimos fue justamente juntarnos entre varias instituciones, el CONICET, la Universidad Nacional de Tucumán, la UNAM, el INTI, para pedir este subsidio. Entonces, la idea es tener una red de plantas piloto, ¿sí?, que puedan estar coordinadas para hacer trabajos científicos de transferencia, de capacitación, etcétera, siendo las diferentes plantas piloto de diferentes tipos. Y están distribuidas geográficamente. Estas son las instituciones que les estaba diciendo. Estos son los institutos que participan, entre ellos el nuestro. Y bueno, ahí está en el mapa de Argentina, ven allá estamos nosotros, aquel bracito allá es Misiones y nuestro partner están en Tucumán. Entonces, la idea es que tenemos varias plantas piloto que ya están instaladas en Tucumán y que lo que se va a ser incrementarlas en su capacidad y para nosotros va a ser crear una planta piloto, que es lo que nosotros llamamos biorefinerías del NEA. Bueno, estas son las actividades que les decía que vamos a hacer. Vamos a tener dentro de lo que es el gobierno un consejo consultor que va a tener miembros del gobierno y de la industria. Y esto es lo que tenemos. 2 plantas piloto de Proimi en Tucumán, una de Itanoa en Tucumán, una en la Universidad de Tucumán, 4 en el INTI, que es el Instituto Nacional de Tecnología Industrial. Esto es lo que hay en Tucumán, que ya hay. Y lo que vamos a hacer nosotros es una planta piloto para nosotros y un laboratorio analítico para el INTI. Entonces así surge lo que llamamos, como les decía, biorefinerías del NEA, donde la idea nuestra es poder hacer una estructura de biorefinería que sirva para múltiples materias primas y ser usada para obtener múltiples productos. O sea que no es una planta piloto de uno, sino de varios productos. Esta es la idea, ¿no?, sobre la base de la biomasa, obtener diferentes productos de las diferentes fracciones, como ácido lebulínico, fórmico, furfural, alcohol furfurílico, ácido acético, xilitol, fibra de carbono, nanocelulosa, ácido láctico, butanol, ácido succínico, por decir algunas. Bien, entonces, para terminar, puse una frase que me pareció interesante de Stora Enso, de un directivo de Stora Enso, que es una gran empresa multinacional de pulpa y papel, que dice: estamos trabajando hacia ser una compañía de materiales renovables en vez de una compañía de papel. La biomasa se da, pero hacer biomateriales es una decisión estratégica. Entonces, en vez de quemar todo, podemos crear valor a partir de ello. Acá están mis datos por si los quieren, si quieren escribir o nos quieren visitar, y muchas gracias.
Thank you, Cristina, for your nice presentation. We have time only for 2 questions. Question? I think all is clear here. Some questions? Marilo.
Primero felicitar.
Gracias.
Que ha presentado Lo que nos parece a veces tan obvio y a lo mejor tan árido, y presentarlo de una manera, ¿no es cierto?, que es atractiva y moderno. O sea, la mirada de hoy, obviamente, la estás haciendo completamente tú con estos nuevos materiales. Y te quería preguntar, ¿no es cierto?, si yo sé que tú has contado recientemente un gran esfuerzo para hacer estas plantas piloto, ¿ya? Y ¿ha sido difícil en Argentina implementar la industria, la nueva industria? Porque en Chile no hay industria prácticamente. Entonces, por eso te pregunto, ¿cómo ha sido el camino, la nueva industria, la bioindustria?
Bueno, por supuesto que es difícil, está siendo difícil, pero lo que pasa es que hay muchos incentivos económicos por parte del gobierno. O sea, sí, se ha creado, por ejemplo, yo soy miembro de la COBIOMAT, que es una comisión de biomateriales, donde se otorga a las empresas, a las pymes que trabajan con biomateriales, un sello que se llama Sello de Bioproducto Argentino. Entonces, eso le da un atractivo como para poder exportar, por ejemplo. Y bueno, y así como eso, créditos, subsidios, apoyos para investigación, apoyos para hacer investigaciones conjuntas entre empresas y grupos de investigación. Así que, bueno, el año pasado se promulgó la ley de promoción de la ciencia, con lo cual se está aumentando año a año el dinero destinado a eso. Bueno, yo no les mencioné, pero esta planta piloto, este CITES que nosotros presentamos y nos aprobaron, iba a ser financiado por el BID, pero luego de evaluar el proyecto dijeron que era estratégico para el país y entonces se iba a financiar con fondos del Estado. Así que realmente el Estado está poniendo mucho para este tipo de cosas.
En esto.
Sí.
Qué bien, gracias.
Thank you, Cristina. Other question? Okay, la otra Cristina.
Muchas gracias. Muchas gracias, María Cristina. ¿No? Ok. Bueno, yo creo que la diferencia de lo que tú presentas, por lo que yo observo, nosotros hemos trabajado muchos años también asociados a industria celulosa, pero por lo que tú presentas, el problema fue al revés. Es decir, la empresa se vio que tenía un problema, como presentó Denise ayer también, y buscó la solución. ¿Ya? Es distinto cuando desde la academia tú quieres proponer soluciones, porque los esquemas, por ejemplo, de los productos que tú puedes obtener a raíz de un árbol son archiconocidos en los libros y en todos los lados. Entonces, el tema es, si la industria tiene un problema y lo quiere solucionar, la cosa camina. Pero si la universidad piensa que tiene que darle solución a la industria, la cosa no camina. Entonces, yo creo que es nuestra estrategia la que tenemos que cambiar, ¿ya? Y lo otro que tú mencionas también ahí, por ejemplo, la creación de estos centros que son interdisciplinarios, es súper importante, porque nosotros tenemos la manía de ser muy individualistas en nuestra investigación, avanzando en ese sentido, y ojalá nosotros también lo pudiéramos implementar y copiar, porque encuentro que es una excelente iniciativa. Así que muchas felicitaciones.
Bueno, muchas gracias. No, cuando, por ejemplo, yo presenté ante el CONICET, que fue el organismo donde yo trabajo, ante miembros del CONICET, del Ministerio de Ciencia y Tecnología, me hicieron presentar el proyecto. Yo les decía que estaba buscando justamente hacer un consorcio público-privado. Y entonces ahí la presidenta del CONICET me dijo, no, me dijo, tiene que ser público-público, porque si uno lo hace público-privado, la empresa se quiere quedar con todo porque pone un poquito y después se cree que es dueña de todo lo que se hace. Entonces, por eso ellos generaron este programa público-público, donde son todas empresas son todas instituciones del Estado las que pueden participar, al menos 3. Por eso nosotros pusimos el INTI, las universidades y el CONICET, por ejemplo, en nuestro caso, ¿no? Y bueno, y eso que vos decías de la industria lo estábamos hablando ayer con Mara, ¿no? Que hay dos extremos, o decir, bueno, yo tengo que esperar que la industria me pida todo y tengo que responder a la necesidad de la industria. Y lo otro es decir, bueno, no, yo respondo a necesidades de la industria, pero también creo cosas vanguardistas, porque tampoco puedo estar corriendo, para correr atrás de la necesidad de una fábrica, tienen su laboratorio de desarrollo. ¿Sí? Nosotros estamos un poco más para otra cosa. Entonces, bueno, sí, trabajamos con ellos, les proponemos cosas. Ahora, por ejemplo, ¿por qué no hemos podido avanzar con la aplicación? Porque nuestra idea es hacer una segunda etapa de aplicación de nanocelulosa en la máquina de papel directamente. ¿Y por qué no lo hemos podido hacer? Porque sacamos la cuenta que con nuestro equipito necesitamos un mes de trabajar 8 horas por día para tener un día de aplicación en la fábrica. Y en un día de aplicación de la fábrica, por ahí ni llegamos al estado estacionario, como para poder evaluar resultados. Entonces necesitamos tener la instalación piloto para avanzar sí o sí, ¿no? Pero bueno, ahí, digamos, nosotros lo que hicimos fue mostrarle a la fábrica lo que podían lograr si hacían una cierta innovación, ¿sí? Porque las empresas, por lo menos lo que son las forestales y las papeleras, son muy tradicionales. Es muy difícil hacerlas innovar. Entonces hay que mostrarles esas posibilidades para que se entusiasmen.
Gracias. Aquí nosotros tenemos un ejemplo en general con la celulosa Arauco y Constitución, que ellos tienen su propio centro de investigación que es Bioforest. Entonces nosotros podemos interactuar con Bioforest, pero siempre asociado a la necesidad que ellos tienen de resolver un problema. Entonces nosotros tenemos que mostrar las capacidades que existen en las diferentes áreas y en las diferentes instituciones. Y ahí a veces donde fallamos también.
Es un poco, claro, es un poco complicado. Sí, con Arauco es complicado. Nosotros tenemos Arauco en Misiones también. Es una de las grandes fábricas, sí. Pero bueno, siempre hay otras vías, ¿no?, de entrar. Bueno, muchísimas gracias, ¿eh?
Okay, thank you very much for those presentations. Now we have time for a coffee break, and we will start the next round of talks in 20 minutes? 20 minutes. Okay, see you.
Okay. Go ahead, please.
Bueno, buenos días a todos. Vamos a continuar con el tema de biorefinería. Y lo que he preparado, digamos, es ver un poquito los tipos de fraccionamiento que se utilizan para tratar los materiales lignocelulósicos referidos a la plataforma de bioquímica o de base azúcar. También pertenezco al Instituto de Materiales de Misiones. Bueno, entonces podemos decir como introducción que la biomasa lignocelulósica representa un recurso sostenible de bajo costo que puede ser convertido en combustibles y productos químicos en gran escala. También es atractiva porque podemos desarrollar nuevos productos con otras funcionalidades. El fraccionamiento es considerado el paso más importante para obtener una conversión eficiente en productos de alto valor. Pero para esto se requiere el desarrollo de procesos económicos y robustos para un fraccionamiento efectivo en los constituyentes de la biomasa celulósica, los constituyentes principales como la celulosa, hemicelulosa y lignina. Originalmente, el término fraccionamiento es equivalente a pretratamiento. En el tratamiento de la biomasa originalmente se utilizó el término pretratamiento porque era una etapa previa a convertir estos materiales más fáciles de convertirse en azúcares a través de una hidrólisis enzimática. Es decir, eran tratamientos suaves. Posteriormente los tratamientos fueron aumentando un poquito más la complejidad, digamos, y entonces se vio la necesidad ya de separar otras fracciones de valor, por ejemplo, la lignina. Entonces, podemos decir que la función del fraccionamiento es tratar de recuperar de la biomasa lignocelulósica los componentes principales, tratando de efectuar el menor daño posible para que sean poder tratarlos como productos de alto valor. Por lo tanto, el objetivo del fraccionamiento es principalmente deconstruir la estructura tridimensional de la biomasa lignocelulósica, incrementar el área superficial, la porosidad del material, es decir, permitir o tratar de obtener una fracción de celulosa fácilmente hidrolizable, modificando el grado de polimerización y también reduciendo la cristalinidad. Tratando de que esos procesos requieran de la mínima cantidad de productos químicos, energía, agua, y también utilizar equipos de bajo costo, tamaño y simples. Lógicamente, nosotros aplicamos, digamos, en esta estrategia de fraccionamiento mucho lo que hemos aprendido, digamos, de las fábricas de pulpa, que realmente son, yo digo, las primeras biorefinerías. Por lo tanto, también, digamos, el objetivo en lo que es la plataforma de azúcar es proveer altos rendimientos de hidrólisis enzimática. Cuando hablamos de alto rendimiento, tendríamos que llegar a conversiones de celulosa disponible mayores del 90%. Minimizar, otro objetivo es minimizar productos inhibidores de la fermentación, como son el ácido acético, el furfural y el hidroximetilfurfural, que provienen, digamos, de la degradación de los azúcares, y evitar también la incorporación de compuestos fenólicos que vienen de la lignina. Y bueno, también permitir la recuperación de hemicelulosas y lignina como coproductos, digamos, del proceso. Y por lo tanto, también, digamos, otro objetivo es tratar de que ese pretratamiento requiera mínimas etapas posteriores, como por ejemplo lavado o neutralización. Bueno, ya vieron ustedes la complejidad, digamos, de los materiales lignocelulósicos. Sabemos, digamos, los componentes principales estructurales, que son la celulosa, hemicelulosas y lignina. También tenemos componentes, productos del metabolismo de los materiales lignocelulósicos, como son los extractivos, inorgánicos. El objetivo del pretratamiento es tratar, digamos, de romper esa estructura tridimensional. Originalmente es para, digamos, facilitar el acceso de la enzima para la hidrólisis enzimática. Bueno, ya vieron en las presentaciones anteriores más o menos el rango de estos componentes, podemos decir en términos promedio entre 50 y 60% de carbohidratos, 20-30% de lignina. Y además de extractivos y cenizas. Aquí tenemos, hoy día, digamos, los pretratamientos que encontramos en la bibliografía y podemos decir que más o menos tienden a infinito. Pero bueno, estos serían los más usuales, en los que algunos están más desarrollados. Por ejemplo, el pretratamiento con ácidos diluidos, el objetivo, digamos, la principal acción, digamos, es la hidrólisis de las hemicelulosas, es decir, aumentar, digamos, la porosidad de ese material a través de la extracción de hemicelulosas, ya que queremos hidrolizar el material sólido. Se utilizan, digamos, generalmente ácido sulfúrico, ácido fosfórico y otros ácidos fuertes. Los pretratamientos alcalinos utilizan hidróxido de sodio, hidróxido de calcio, carbonato de sodio. El tratamiento, en el caso de los tratamientos alcalinos ya el modo de acción es que ya extraemos lignina. En el tratamiento de explosión de vapor, que utilizamos vapor a alta temperatura, podemos utilizar algún catalizador. También hacemos la hidrólisis de la hemicelulosa y una desestructuración de el material lignocelulósico. También tenemos los líquidos iónicos, solventes eutécticos profundos, cuya función es fraccionamiento de los polímeros, la acción, digamos. Los procesos organosol, al igual que los alcalinos, también extraen bastante lignina. Bueno, también la reducción del tamaño de partícula por medio de la molienda nos va a incrementar, digamos, la acción o el rendimiento de la hidrólisis enzimática, y también los tratamientos biológicos menos utilizados precisamente por los tiempos que requiere el tratamiento, sobre todo con hongos, para llegar, digamos, a una estructura lignocelulósica fácilmente atacable por las enzimas. Bueno, acá tenemos en este gráfico un poquito la composición del material sólido en función del tipo de pretratamiento. Acá tenemos un pH ácido, alrededor de 2, y pues, en otro extremo tenemos un tratamiento alcalino. Lógicamente los tratamientos ácidos no van a retirar la mayor parte de las hemicelulosas, como tratamientos ácidos, autohidrólisis o explosión de vapor. Ya acá tenemos un pH ligeramente alcalino en este proceso AFEC, que ya lo vamos a ver. Ya acá también tenemos una reducción del contenido de lignina, después oxidación húmeda. Y aquí tenemos un proceso netamente alcalino, en donde vemos que en el material sólido vamos a tener las hemicelulosas y la celulosa y se retira la lignina. Vamos a hablar sobre estos métodos de pretratamiento, con algunos ejemplos de trabajos que realizamos en nuestro laboratorio. Empezando con los procesos hidrotérmicos, llamados también de autohidrólisis o explosión de vapor. El tratamiento de autohidrólisis trabaja solamente con agua caliente, temperatura 150, 230 grados centígrados. Tenemos que utilizar altas temperaturas porque debajo de 100 grados no tenemos efectos hidrolíticos. Por encima de los 220 grados ya vamos a tener algo de degradación de la celulosa. Esa sería la temperatura límite, porque por encima de 240 grados ya tenemos reacciones de pirólisis. El tiempo de reacción en los tratamientos hidrotérmicos varía desde segundos a horas y depende de la operación, condiciones de la temperatura de operación. Estos tratamientos hidrotérmicos están muy bien estudiados, o sea, se generó este factor de severidad que combina las variables tiempo y temperatura. Es decir, que nosotros, en teoría, podemos trabajar a distintos tiempos, distintas temperaturas, obtener un mismo factor de severidad. Eso nos da que vamos a tener en ese material el mismo efecto hidrolítico, sobre todo de las hemicelulosas. Si nosotros a estos tratamientos se le agrega algún catalizador ácido, se puede utilizar como herramienta de control del proceso el factor de severidad combinado. En los tratamientos hidrotérmicos se trabaja con una de las variables, la relación sólida a la concentración de sólidos. Los valores, ustedes saben que la biomasa, los residuos de aserradero tienen baja densidad estéreo, entonces, digamos, necesitamos utilizar altas relaciones líquido-sólido, alrededor de 10. Se pueden encontrar de 2 hasta 100 en la bibliografía. Pero altas relaciones líquido-sólido están asociadas con reactores continuos, mientras que las bajas relaciones están asociadas con los tratamientos con vapor. ¿Qué pasa? Es muy importante esta variable porque nos va a determinar también en gran medida la severidad del tratamiento, porque lógicamente lo primero que nosotros vamos a liberar en el tratamiento hidrotérmico son los hidrólisis de los acetilos. La hidrólisis de los acetilos nos produce ácido acético. Como consecuencia, se va a acidificar el medio. Disminuyendo la relación sólido-líquido, aumentamos la concentración, y eso ácida, digamos, del medio, y eso va a favorecer la depolimerización. Pero si nosotros reducimos la relación, podemos tener un efecto negativo en el sentido de que podemos tener una capacidad amortiguadora del pH, es decir, no nos va a bajar mucho el pH, precisamente por el contenido de inorgánicos del material. Bueno, entonces, la autohidrólisis utiliza temperaturas del orden de 200 grados centígrados, no se requeriría catalizador, sobre todo en un tipo de materia prima que son las latifoliadas y los residuos, las gramíneas, que tienen alto contenido de acetilos. Ya cuando nosotros estamos trabajando con coníferas, sí tenemos que agregar algo de ácido. Entonces, en la autohidrólisis tenemos una alta recuperación de hemicelulosa, pero no tenemos solubilización de lignina. La fracción líquida está sin compuestos inhibidores por la baja degradación de los azúcares. Por lo tanto, las hemicelulosas están principalmente como polímeros de pequeño tamaño, oligosacáridos. Bueno, ¿cómo funciona la autohidrólisis? Bueno, es catalizada por los iones hidróneo proveniente de la ionización del agua. Esto también, la concentración de iones hidróneos va a aumentar si nosotros tenemos ácido acético en el medio, mejorando la cinética de la reacción. La depolimerización de las hemicelulosas se realiza por la hidrólisis de los enlaces glucosídicos y también tenemos hidrólisis de los grupos acetilos. Por lo tanto, vamos a tener en la fracción líquida una mezcla de oligosacáridos, monosacáridos, ácido acético, derivados de productos de descomposición en baja proporción, pero furfural de los azúcares, furfural, hidroximetilfurfural, los cuales se pueden también descomponer en ácido fórmico y en el caso del hidroximetilfurfural se puede descomponer a lebulínico. Bueno, este es un ejemplo de pretratamiento con autohidrólisis de bagazo de caña de azúcar, ya se lo mostró Cristina, en donde sí tenemos que hacer una poshidrólisis, ya que tenemos la mayoría, el principal componente de las hemicelulosas del bagazo son las los silanos, perdón. Entonces, como obtenemos oligosacáridos, tenemos que hacer una poshidrólisis y luego tenemos que sacar el ácido sulfúrico precipitándolo con hidróxido de calcio, hacer un tratamiento de eliminar algunos compuestos aromáticos, los grupos ácidos y el hidroximetilfurfural y furfural por ejemplo, con carbón activado, los ácidos con unas resinas de intercambio aniónico para recién poder pasar a la fermentación para producir xilitol. En el tratamiento de explosión de vapor se utiliza vapor a alta presión, 20-50 bares entre 210 290 grados centígrados, pero por muy corto tiempo, segundo, algunos minutos. El vapor es condensado a alta presión, es decir, va a mojar el material, pero luego, cuando se produce el soplado, porque la explosión de vapor queda muy poco tiempo, luego se hace una despresurización brusca, entonces la evaporación de esa humedad genera la desestructuración del material. Por lo tanto, genera, por supuesto, la rotura de enlaces intramoleculares e intermoleculares. Este es un tratamiento de fraccionamiento en donde realizado sobre acerrín de pino, en donde sí realizamos una impregnación con ácido sulfúrico. Probamos dos estrategias, también hicimos una desresinación. Ustedes saben, los pinos tienen un elevado contenido de resinas, y luego se sometió a una explosión de vapor y de ahí a una hidrólisis enzimática. Digamos que en este trabajo la conclusión es que este sistema genera muy bajos rendimientos de glucosa. El pino es un material bastante recalcitrante en lo que hace a la hidrólisis enzimática, por lo tanto, digamos, se requieren otro tipo de tratamientos, por ejemplo, tratamientos deslignificantes. Una modificación del tratamiento hidrotérmico es la oxidación húmeda. Acá se utiliza, digamos, se incorpora además de la temperatura, oxígeno y aire como catalizadores, aunque se está utilizando actualmente como catalizador el carbonato de sodio, generando condiciones moderadas y alcalinas. Esto genera una alta conversión de la biomasa en monosacáridos con baja formación de aldehídos furánicos y fenólicos. Pero la presencia de álcalis, lógicamente, genera un incremento de los ácidos carboxílicos. En cuanto a el otro tipo de pretratamiento, la hidrólisis ácida, se utilizan dos enfoques. Uno es utilizar ácidos concentrados baja temperatura o ácidos diluidos alta temperatura. Cuando hablamos de ácidos concentrados y baja temperatura, como ácidos utilizamos ácido sulfúrico, nítrico, clorhídrico o trifluoracético, muy altas concentraciones, 72% de ácido sulfúrico, 41% de clorhídrico, 100% de tetrahidrofurano. Trifluoracético, perdón. Bueno, este proceso es más caro y tiene muchos problemas de corrosión. Remueve no solo celulosa, hemicelulosa, sino también celulosa, y queda un residuo insoluble enriquecido en lignina. Pero, digamos, la operación a baja o media temperatura genera pequeñas cantidades de productos de degradación. Cuando hablamos de procesos ácidos diluidos, estamos hablando también más o menos de los mismos ácidos, pero ya en concentraciones, estos ya referidos a material seco de 0,5 o 1,5%, y temperaturas de 121 a 160 grados, dependiendo del tipo de material lignocelulósico que se esté utilizando. Por lo tanto, se alcanza altos rendimientos de azúcar a partir de hemicelulosa, también generan baja cantidad de productos de degradación al utilizar baja concentración de ácido, y tienen pocos problemas de corrosión. Una alternativa es agregar, digamos, al sistema anhidro carbónico, que va a formar ácido carbónico. Es un método alternativo dentro de los tratamientos ácidos. Es decir, se utiliza en condiciones suaves de pH de hidrólisis. Un método patentado, digamos, de tratamiento se puede colocar dentro de de esta clasificación de hidrólisis ácida es el método SPORL. Usa una solución de sulfito que puede ser de sodio, magnesio o calcio, trabaja a un pH 2 a 4 por alrededor de 30 minutos, temperatura 160, 180, y luego ese material es desfibrado con un molino de discos. Pero este proceso logra rendimientos de glucosa en la hidrólisis enzimática del orden de 88% de glucosa masilosa y un 32% de remoción de lignina. Bueno, otro método, digamos, que se presenta como viable económicamente, como una estrategia viable, son los métodos alcalinos. La ventaja de los métodos alcalinos es que sí, acá vamos a tener solubilización de la lignina, con baja solubilización de celulosas y hemicelulosas. Esto va a generar, al eliminar lignina, un incremento de la digestibilidad de la celulosa. Bueno, se pueden dividir en dos grandes grupos, si se trata con hidróxido de sodio, hidróxido de potasio o calcio, o si se utiliza amoníaco. Las cargas, digamos, que se utilizan cuando se trata con hidróxido de sodio son bajas, de 5 a 15% sobre biomasa seca. Es uno de los métodos más estudiados, muy efectivo para incrementar la digestibilidad de las latifoliadas o los residuos agrícolas que tienen bajo contenido de lignina. La temperatura es un factor importante. Trabaja, digamos, con bajas temperaturas y presiones, inclusive también se pueden utilizar condiciones ambientales. Un ejemplo de fraccionamiento alcalino, digamos, es el proceso de pulpado Kraft en la industria de la celulosa. Como les decía, la fábrica de celulosa Kraft es un ejemplo de biorefinería. Hoy día se utilizan estrategias para la extracción, una preextracción de hemicelulosas, para producir bioetanol, químicos y polímeros. Esa pulpa pasa al proceso de pulpado, sobre todo cuando se fabrican pulpas para disolver. Y bueno, acá seguimos el camino habitual que tiene una fábrica de celulosa, es decir, obtener energía a partir de el licor negro. Se puede retirar una fracción de lignina para producir ligninas técnicas, también con objetivo de producir químicos y polímeros. Bueno, acá tenemos otro ejemplo de tratamiento de biomasa con álcali, en donde al final del proceso vamos a obtener un residuo sólido rico en celulosa. Podemos acidificar hasta pH 5 y precipitar con etanol las hemicelulosas. Y ese licor residual que nos queda se puede concentrar y luego llevar a pH 2 para separar la lignina. Bueno, una variante, digamos, que se utiliza para mejorar la digestibilidad de los materiales lignocelulósicos, sobre todo que provienen de la explosión de vapor y tratamiento hidrotérmico, es hacer un tratamiento adicional con peróxido de hidrógeno, utilizando pequeñas cantidades de 1 a 3%, tiempo de 45 a 5 horas, temperaturas bajas, y esto es lo más importante, pH 11,5. Lógicamente, esto aumenta la digestibilidad de los materiales tratados hidrotérmicamente o con explosión de vapor, pero no tenemos no nos aumenta la eliminación de lignina. Este es otro ejemplo de método de explosión, de método alcalino, que es el proceso AFEX, que utiliza álcali y el proceso de explosión de vapor. Consiste en impregnar, digamos, el material lignocelulósico con amoníaco anhidro líquido, trabajar a temperaturas moderadas, altas presiones por pocos minutos. Por lo tanto, tenemos aquí una combinación de efectos físicos dados por la explosión de vapor y químicos por el tratamiento con amoníaco. Esto genera la rotura de los enlaces del complejo lignina-carbohidrato, la hidrólisis de la hemicelulosa y la decristalización de la celulosa. Otro método de pretratamiento es el tratamiento organosol. También es, al igual que el álcali, va a remover lignina y hemicelulosas y utiliza solventes orgánicos como etanol, acetona y algunos ácidos orgánicos como fórmico o ácido acético. Recientemente se está investigando la utilización en el fraccionamiento de otros solventes como la gamma-valerolactona, la metil-isobutilcetona, el tetrahidrofurano o el 2-metil-tetrahidrofurano. Aunque el sistema más estudiado es las mezclas etanol-agua, que puede tener o no un catalizador, ácido o alcalino. Se me fue la... Bueno, enseguida. ¿No tengo más nada de tiempo? No vi el amarillo. Bien, ok, vamos a acelerar. Bueno, estas son las condiciones de tratamiento de etanol-agua, algunos ejemplos utilizados en biorefinería. Nosotros utilizamos o comparamos el efecto del proceso soda-etanol en la etapa de deslignificación, obteniendo resultados muy comparables con el proceso Kraft y soda-antraquinona cuando se utiliza acerrín. Bien. Bueno, acá se ve, digamos, el efecto de la presencia de álcali en la digestibilidad. Lógicamente, aumentando la concentración de álcali, disminuye, digamos, la cantidad de lignina, aumenta la digestibilidad del material. Bueno, también estuvimos trabajando con gamma-valerolactona agua. Vamos a dejar los líquidos iónicos, que son, la verdad, no los que hemos estudiado en nuestro laboratorio. Y voy a las conclusiones, nomás. Entonces, los métodos de fraccionamiento de residuos son diferentes a los métodos de pulpado, tienen que ser diferentes a los métodos de pulpado tradicional, ya que requiere recuperar todos los constituyentes del material, y es difícil desarrollar un proceso general para todo tipo de material, porque esos son muy diferentes. Por ejemplo, vimos que el pino es un material muy recalcitrante, digamos, a la hora de la hidrólisis enzimática. Por lo tanto, la selección del pretratamiento adecuado depende, lógicamente, de la aplicación final. Bueno, disculpen por haberme excedido en el tiempo. Bueno, muchas gracias por la atención.
Thank you very much.
Gracias.
We make an applause for the Professor Felicia, and we ask the public if they can ask questions later in the lunchtime. and welcome to Professor Dr. Jakub Kusiakow from the University of Göttingen. The Professor Kusiakow will present hotspot and hot moments in soils. It's a change in the program, but it's more abroad.
Yes. Hello, everybody. And because lunch is coming soon, I would like to make a short refreshment, and therefore I change the topic for a little bit more exciting, and because the most of the community here is more or less related to microbial life. So we will speak about hotspots and hot moments by microorganisms. This talk is based on a paper which we made together with my colleague and friend, Evgenia Blagorodskaya, a couple of years ago. And in this talk, we raised a couple very important questions. At first, we raised the importance of heterogeneity of properties in soil. And the second is that we need not only the data, we need also concepts. And if you look on soils, you would see some spatial structure. This spatial structure I presented here based on 4 pictures, just on the soil surface. And I think generally in all ecosystems, the spatial structure depends on the limiting factor and the redistribution of the limiting factor. Initially, this redistribution is ongoing by abiotic processes. And later, the biota increase this structuring and make some environment which have some spatial pattern. This spatial pattern can be seen here, for example, because of oxygen limitation, limitation of nutrients in tundra, limitation of salt-free water in dry steppe, or limitation in water in very dry steppe by Gilgai relief. This we can see on the soil surface. But what about the limitation for soil microorganisms, for microorganisms in soil, for microbial activities? Actually, microorganisms can be limited by water, by oxygen, by nutrients, by toxicity of some substances. But nearly in all soils, microorganisms are limited by available substrates, by food. So the microorganisms in soil have not so good life like microorganisms in cultures, in nutrient culture. And there are many papers about. And I think if this factor, limitation by by available carbon and energy, will be redistributed, then it's coming to the structuring of the soil. What are the locations with plenty of available carbon? I think there are 4 main locations. This is detritosphere, so the soil volume around the decomposing plant residues. This is a rhizosphere, the soil around the living roots, the biomes which were made by roots or by some soil animals, and also aggregate surface when organic, dissolved organic material will be migrated together with water. These 2, detritosphere and rhizosphere, are actually the primary sources of carbon in soil. These 2, biopores and aggregate surfaces, they are mainly a redistribution of these primary sources, and they are mainly important in the subsoil. And I would like to show in some next slide that in these locations, in this detritosphere, rhizosphere, and some biopores, the local input of available carbon is sometimes 10 or even 100 times higher compared to the bulk soil. And therefore, in these locations, some hotspots will be formed. I will mainly speak about the rhizosphere, but I think some of the processes are similar in other locations too. So our concept of hotspots is that the hotspots are small soil volumes with much faster process rates and very intensive interactions compared to the average soil conditions. It means if we relate the process rates to the soil volume, probably we will have such non-uniform distribution, in which the most part of the soil is some kind of dead soil. I'm sure that many soil scientists would like to kill me because of this word. Yeah. Yeah. But I would like to show you the reverse, that there are some locations in which the processes are much faster. And usually when we collect the soil, we mix the soil. And then we are getting the average, the mean, which is very close to the dead soil because of its huge volume. But actually, again, I would like to show you some examples that despite the relative volume of the hotspots, it's 10 to 100 times smaller than of dead soil, the process rates there are 10 to 100 times faster. And my question to you, maybe you know from your life, some hotspots. Do you have any idea? I have just a couple of good examples. You see here the impact factor of more or less good journals, Nature and Science, and you see that the impact factor here is about 38, and here about 35, and this is the distribution of the citations of the papers in the journals. If we take the median on the scientific, it will be just about 15 or here about 13, but actually this high impact factor, nowadays it's much higher, this is just for 2015 or so on, will be made by few papers, which citations are much higher compared to the median. So these hot papers make the impact. Another example, which probably we also know well, so the location of the people in the world. There are also some hot locations around the world, what we can see here during the nighttime. And I think such locations bring the communities of microorganisms together because they have high availability of carbon and energy, abundance and activities of microorganisms, turnover rates, weathering, and some others. And just one picture about the abundance of microorganisms on the root tip, living and dead microorganisms. And this is clear because it's preferred habitat for microorganisms. And I would like to show you some examples, mainly on the rhizosphere, mainly related to the microorganism and especially of the activities and functions of the microorganisms in the rhizosphere. One very simple example how we can measure the microbial growth rates in soil. We just add very available carbon, for example, glucose, and measure the respiration over a few hours. And you see here two lines, one line blue for root-free soil and green for rhizosphere soil. And based on this exponential increase, we can calculate the microbial growth rates. It looks not very big difference, just about 25%. But these are 25% per hour. It means in 2 hours, it will be 50%, in 4 hours, more than double. It means the growth rates are much higher in the rhizosphere compared to the root-free soil. Another example, mineralization potential, means how much food microorganisms can utilize per unit of time. And this is reflected by the, here in this case, we added only one excess amount of glucose, and here we add various increasing amounts of glucose and measure the respiration. You see that the respiration in the rhizosphere, green line, is about 2.5 times higher than in the root-free soil. It means per time unit, microorganisms in the rhizosphere can utilize about 2.5 times more food. It means in the hotspots, we have higher carbon input, for example, from roots, and much faster mineralization, and this leads to faster turnover of microorganisms and consequently also to strong increase the fluxes. but no changes of the pools. And please have in mind that in most of our studies, we measure the pools and not the fluxes. Let us look for some other functions. Here we collected some various functions and densities of the parameters in rhizosphere and in detritosphere compared to the bulk soil. If the values are about 1, then these are the same like in the bulk soil. If the values are 2, 3, and so on, then they are 2, 3, or more times higher than in the rhizosphere. And on the y-axis are in blue presented static parameters like concentration or abundance, and in red are presented dynamic parameters like enzyme activities. And actually we expected that the dynamic parameters will be much stronger expressed in rhizosphere and detritosphere compared to static, but this was not the case. But nevertheless, we see that nearly all parameters that we have tried to collect from literature are actually much stronger, 2, 3, 4 times in the rhizosphere compared to the detritosphere. Let us look now for localization. So how we can localize some activities and some function. One of the options is that we label the plants in the atmosphere with the shifted isotopic composition, for example, with radioactive 14 CO2. The plants cannot differentiate various isotopes, and they bring the carbon into the soil, and we can measure this in various soil pools, in shoots, roots, soil organic matter, microorganisms, and also in respiration. Let us look for the rhizodeposition, for the localization of rhizodeposition. You see here the roots of a ryegrass, and this ryegrass was labeled before, just 6 hours before, in such a box, which I presented before. This picture we can see by eyes. This picture is an autoradiography. Autoradiography is very, very old method, much, much older than even me, in which the activity of radioisotope will be impressed in the photographic paper. Not nowadays digital photography, but old method of the photography on paper. On the first glance, both pictures look similar. However, if we look more precisely, we see that actually the dark color, black color, is presented mainly on the root tips. Not only here, you see it on many other places too. And from other studies, from the balance studies, we know that this amount corresponds to about 5 to 15% of totally assimilated carbon, or 10 to 25% of net assimilated carbon. And consequently, and if we look on the area is maybe just 5% of such root tips, maybe even less. This is, again, very old method. Nobody uses it nowadays. We use another approaches, similar by imaging techniques, which is like digital photography. You see here the roots of maize growing in sand, not really soil. And maize was labeled similarly in the 14C atmosphere as I presented you before. And then we can make such picture of the localization of 14C. And you see the red color mainly on the root tips, which you cannot recognize even here, these root tips. They take about 10 to 25% of net assimilated carbon, and actually, these are the locations where the microorganisms are happy. It is clear that this carbon will be located on the locations of root growth. Therefore, in the next slide, we very carefully removed these roots, but you don't see here the roots more, but nevertheless, you see very similar localization of organic carbon. It means this organic carbon was released into the soil, and again, will be used or can be used for microorganisms. If you look again on the area of this picture, maybe these locations, these hotspots, take just about 1% of the soil volume. What about other hotspots? I presented you the release of carbon into the soil. Let us look for the other hotspots. This is water uptake by plants made by another group of Andrea Calminati. Let us look on this picture. Please have in mind the color are reverse. It means blue color correspond to dry conditions, white color correspond to the water flux. This is not patients of maximal water uptake by the roots. And this is under dry conditions. When the soil was wetted, irrigated, then this location, these hotspots of water uptake remains. Let us look further for some other hotspots. Oxygen uptake, pH changes close to the root surface. Another option, this is not song, this is indicator in agar-agar, but nevertheless you see very good, very clearly acidification of volume close to the roots and close to the root tips. And according to the assessment, it is about 2 units pH decrease. 2 units pH decrease means the concentration of H plus is about 100 times higher in such locations. Let us look now, or link now, some various properties. This is one of the picture of carbon release into the soil by the roots, and red color correspond to the maximal activities, or maximal activities of the response. But we can try to look how this carbon will be used by other functions, for example, by microorganisms, and for the same root, it is presented activity of acid phosphatase. Acid phosphatase is one enzyme which break organic phosphorus to the phosphate, and acid phosphatase can be released by roots and can be produced by roots and by microorganisms both. You see very good correspondence between release of carbon and activity of phosphatase. And on the same roots, we made another activity of alkaline phosphatase. Alkaline phosphatase cannot be produced by roots and can be produced only by microorganisms. And you see exactly reverse situations that on the roots there is no activity at all, and there are only some other locations with very small activities. It means if the roots do the job, probably microorganisms do not repeat the same job. But I would like to focus on this picture, the attention not only on this nice picture, but also again that the area of such activities is maybe just 10% of the whole area. But the activity, if we compare the red or yellow-red color with blue color, is about 10 times higher. Let us look for a similar picture for another enzyme. You see here the roots of maize. Here with white color, the release of carbon into the soil from the labeled maize. And here's the activity of beta-glucosidase. And you can recognize again that the activity of other enzyme here, beta-glucosidase, is also higher on the root tips. And surely we can analyze this, so the activities analyze the volume and the area are. Here's the area of activities about 8%, here's about 10% of the whole area. This picture was done with roots which have a lot of hairs. Let us look for a mutant. The same study, but the roots have no hairs. And then you see a very different picture that actually the area of the activities increase for about 10 times or even 20 times for beta-glucosidase, just because of the absence of root hairs, which clearly show which function have root hairs here. Based on this, we can try to assess the size of the rhizosphere. So how big is the rhizosphere around the roots. We can take some parts of the roots and can try to make the gradients from the roots and estimate the activities from the root. Do you see here 4 pictures for 4 enzymes? Beta-glucosidase is responsible for carbon cycle, cellulose also for carbon, acid phosphatase for phosphorus, leucine aminopeptidase for nitrogen. And on each picture, you see the distance from the root surface and the activity of the enzymes, and you see 3 lines according to the time. 1 week after root, after root reach this location, 4 weeks and 8 weeks. So this is more or less the time. And you see this time, the activity on the root surface increased very strongly. However, and this is surprisingly, the distance of the size of the rhizosphere, the activity of these enzymes from the root surface to the bulk soil remained actually the same. And I think this is a very important conclusion, that the rhizosphere size remains nearly constant, but the gradients increase with time. And this is one of the factors of the stationarity of some processes. We will come to this also a little bit later. Another picture about the effects of chemical and biological processes on the hotspots. You see here 9 pictures. Here, this is the study was done on very strongly pay-limited soil. This is pay-limited soil, and here we added mineral phosphate, calcium phosphate, and organic phosphate phytate. And it was measured acid phosphatase, alkaline phosphatase, and pH changes. It means this is biological effects on hotspots, and these are chemical effects on hotspots. Biologically, the activities between the blue color and red color is about just 2 times according to the scale. But chemically, the pH change about 2 units here under limitation. It means again about 100 times stronger acidification of the soil close to the roots of the lupin. And if we compare this first column, with these 2 where the phosphorus limitation is absent, we can conclude that actually the hotspots are more stronger in the presence of the limitation. So limitation increase the hotspots. And I think it's very interesting thing to investigate the costs to make such hotspots. What are the energy and carbon costs for removal of such limitations? So I would like to summarize some of the results. In the hotspots, microorganisms are under mainly active and growing. Under the dead soil, they are under maintenance and mainly dormant. They have high carbon use efficiency under dormant state and mainly K strategist. In the hotspots, they have low carbon use efficiency and mainly R strategist. I have not stopped a lot on this picture, just trust sometimes. And it means that in hotspots we have high input and high output. It means that in hotspots the fluxes increase very strongly, but the size probably remains the same because input increase and output is increasing. But the turnover of, for example, microorganisms of organic carbon in hotspots increase very strongly, and mean residence time, what is more or less age of the microorganism of carbon, so very strongly decrease. It means in such hotspots which have higher carbon input and faster mineralization, it means we have strong increase of fluxes, faster turnover, but nearly no changes of the pools. And again, in many of our studies, we measure the pools and not the fluxes. And I think if we would like to investigate sensitivity of something to some global change effects or any management effects, we need to look for fluxes. If we would like to look for stability of carbon in soil, then we'll need to look for the pools. And fluxes are responsible more for variability, pools for stationarity. And they both making the spatial structure. To investigate or to present this spatial structure a few years ago, we made also another paper about the rhizosphere shape and size, about the temporal dynamic and spatial stationarity, and have tried to show that the rhizosphere is probably one of the most important hotspots, which has spatial structure. Spatial structure depending on the opposite processes of release of carbon and uptake of various nutrients. So we estimated this, again, based on various approaches of non-destructive imaging, in which we measured the changes of the activities along the routes and also across the routes, and have tried to make such picture of the distribution of enzymes, mucilage, heavy metals, iron and manganese, exudates, pH, various nutrients, some gases too. But I think the most interesting was about the life around the roots. And about the life, to regret, at that time, we did not find a lot of data. Therefore, this picture was just hypothesized. How various microbial groups are distributed on the root surface and with some distances from the root. Very limited data. And before I go to the time, because of very limited data, with a couple of my friends and colleagues from China, we made estimation of the rhizosphere bacterium of structure and function just in this year. And we aimed to generalize, to look what is generally special between various plants in the rhizosphere, what is the community structure of the microorganism, what are networks, response to various factors, and also the functions. And just one slide from this paper may be interesting. You see here communities of microorganisms in the rhizosphere in blue and in yellow in bulk soil. And just some examples, you see, for example, here microorganisms which are nearly acidobacteria not presented in the rhizosphere. They are strongly depleted in the rhizosphere but enriched in the bulk soil. There are some other communities which have similarly, for example, Timonadas, which are also depleted in the rhizosphere but enriched in the rhizosphere. But there are some other communities, like Rhizobiales, which are strongly enriched in the rhizosphere. So we made such overview, which groups are actually depleted or strongly enriched in the rhizosphere, and have tried also in the next step, from the structure to functions, I looked for various functions, about 12 functions, which functions are especially expressed in the rhizosphere. But I would like to go now back to the timing. You have seen this slide already with these hotspots, and I mentioned them in high input of organic carbon. And important is to mention that for microorganisms sitting somewhere here or somewhere here, probably it is short period of time when the available carbon increase. And when the available carbon increase, then the activities of microorganisms also very strongly increase because they have enough food, energy, and can make their functions. Therefore, we term this as hot moments. Hot moments as more or less lifetime as hotspots. And I would like to ask you also again, do you have any ideas about hot moments in your life? Some people are telling, no, not any hot moments in my life. So, but probably this is not what you think. I see how good you are laughing now about hot moments. But this is not what you are thinking now. I will present you one example of really hot moments. You see here a stadium, and you see some people. These are discussing the next match. These are speaking about the weather. These speaking about the money. They speak and do what they will do today evening. So it means there is high biodiversity. But I think biodiversity is not really connected with functions. This is my view. Yes, it is diverse, but no clear function. But in some moments, all microorganisms have the same functions, because this is a huge community is directed in for one special function. They do the same, no matter what is the reason for this hot moment. But the community is necessary. Community which has the same function. How we can measure activities, hot moments. I present you just one picture for that lifetime of hot moments. You see the same picture what I presented before. I presented before just for 6 hours, but we can do the same picture after some period of time. And you see the activity in this case of release of carbon into the soil is, according to the red color, maximal after 6 hours. It is still present after 2 days, but a little bit less and not more located on the root tips. And after 11 days, it's nearly, the red color nearly disappear. So this is more or less this one of the options to measure hot moments. The periods of much faster process rates. So I presented you before one-dimensionally plus one-dimensionally the distribution of release of carbon, pH, oxygen, or various examples of enzyme activities. Then we tried to relate this to the time. It means time plus two-dimensional as presented on the previous picture. And a little bit just in the last year, we have tried to make three-dimensional picture in space. It means we collected biology, chemistry, and physics. So we made such soil columns, collected such soil columns from the soil, made microcomputer tomography, which allowed the localization of the pores, and then cut such soil cores, and in each such cutting we measured activities of the beta-glucosidase. And I forgot to mention that microcomputer tomography allowed to measure not only the pores, but also to estimate the particulate organic matter, more or less organic residues. Then we made three-dimensional picture which present pores, particulate organic matter, and beta-glucosidase. Can somebody who is responsible for the computer just press here in the middle one time? Oh, yes. So you can recognize this blue color, these are pores in the soil. In green, this is particulate organic matter, root residues, plant residues, and the activities of beta-glucosidase. Surprisingly for us, it was no overlap between particulate organic matter and beta-glucosidase, what we expected. So we are still looking for reasons and for the mechanisms why the overlapping is not so good. Is it the problem of the methods, or is it our problem or our views? So let us try now to link hot moments and hotspots. You see here on the x-axis the spatial extent, so the size of something, and on the y-axis the duration of something. And you see here the size of individual bacteria, a little bit larger than 1 micrometer. Fungi can be so very, very long. And you see here also the bacterial lifetime. And I tried to present here the localization according to our pictures, which I presented to you before, the size of rhizosphere, detritosphere, and others. And the first impression is that actually the individuals are not sufficient to initiate hotspots. So individuals are not enough to produce functions on the larger scales. It means all these are built by colonies, not individuals. The things, and this is important to have functions which will be relevant on the next scale, on the next spatial scale. And this is also an important consequence that it makes sense to work in groups, not as individuals, then your results and your functions will be relevant on the next scale and maybe also in the next time. The second important conclusion is the hot moments are short. Just a couple days or few weeks, so use the hot moments. I would like to come a little bit to conclusions about the evolution of hotspots. You see here just visualization of the following group of processes. Initially, some abiotic processes produce some disturbances. Here, for example, just a crack in the soil, which can be produced by drying or wetting, by freezing and thawing, and, for example, Pancho made also a couple of papers in this direction. And then these locations will be filled out by some biota. For example, by growing roots. These growing roots release carbon, and it means for microorganism energy. Some hyphens of mycorrhiza will be produced, and this will be developed actually by rhizosphere and hyposphere. Then the roots starting to die, and then detritosphere will be produced from the rhizosphere. And then these locations later will be occupied by other animals, for example, or other roots, because these locations contain already organic carbon from previous roots or plant residues. They are partly enriched in nutrients, and they have not so very sharp cants like in the beginning, but they are more convenient for life. But the most exciting is that this situation will be repeated again and again. It means the hot moments will be prolonged by changing of the agent. This is one agent, abiotic, this are biotic agent, and this is another biotic agent. And this will be repeated and again and again. So the hotspots in soil very frequently have multiple origins. And they will be repeated in space and time, and this is leading to the structuring of the environment. We can see it well in the subsoil, because in the most agricultural soil we have tillage, and we don't see such spatial structure. But in natural soils and in subsoils, in agricultural soils, we can recognize such spatial structure. I'm coming to conclusions that microbial hotspots are localizations with available carbon and energy inputs. I think the most important of them are rhizosphere and detritosphere, and we define them not by the content, but by the process rates. For rhizosphere, there is very high availability of available carbon and energy, therefore microbial activities are high. Microbial respiration, growth rates, mineralization potential, activities of enzymes, and therefore the decomposition of soil organic matter localized is also very strongly increasing, which are the priming effects. In these locations, microbial metabolism changed very strongly. I have not stopped with some examples on it, but in hotspots, there are growing microorganisms not under maintenance or dormant, and they are mainly strategists. So the rhizosphere are hotspots which are more quasi-stationary. So the roots are growing, but the spatial structure of distribution of nutrients, exudates, some gases remains mainly the same. The extension in the rhizosphere is just few millimeters, but the lifetime is hours to days. And in the rhizosphere, the of these hotspots is mainly depends on the input of organic carbon by roots. In the detritosphere, I have not presented the data for this, but in the detritosphere, the duration depends on the output. It means on the decomposition rates of the plant residues. But because of the persistence of the agents, we are getting some structuring of the environment from which I started actually my talk at the beginning. So in the hotspots, we have high input and fast decomposition. It means this is very strong increase of fluxes of the turnover, but the pools remains nearly the same. Thank you very much for your attention. And I'm open for your questions.
We have time for a few questions.
Make two.
No, I understood the rhizosphere as the hotspot. I understood. But since you looking for the different the evolution of the microbiome, close to the roots change with of course with the distance along, you know? And especially in the roots there are different localization of the different microorganisms also. O sea, es diferente metabolism also, you know. Entonces it's very difficult to, because we are looking for the big the result of the summary of the lot of mechanism, you know. It's very nice, but it's nice you say it's the hotspot, because yes, it's the hotspot. I understood the differences between carbon in, close to the less than 1 millimeter to the rhizosphere, and the bulk is very different relationship. Really, I can make a question because the question is probably how much time do you think it is important when you fertilize, for example? When you fertilize, the reaction is because the microorganism consume the organic carbon very fast. You know? For example, when you put glucose, the one day is the respiration is the function and nothing more. But the photosynthesis activity, for example, continues increasing.
Yes.
You can explain me.
Yes, I can explain you. We have one of the picture how various activities changed, because activities is not only respiration, not only enzyme activities. This is expression of RNA. This is, for example, like you mentioned, activities of enzymes, and we have very frequently one trigger, but the consequence takes much longer than the trigger. This is your example. And I had such picture for this talk because of the time I deleted it. But if you are interested, in the original paper, this picture is presented, how various types of activities change after the, for example, activation by glucose or by root exudates or something similar. But I would like especially focus on your first part, which was not as a question. You mentioned there is variability along the roots. There is variability between various microbial groups and so on. We have a lot of variabilities, everything. But I think one important part of our science to find rules within variability. And I try to present the rules, not the variability.
Thank you. We can discuss in the university after.
Yes, with pleasure.
So thank you very much. You may guess that I also would like to add the mycosphere as a hotspot. But I've another question to that. So we also try to define actually the extent of the hyposphere, and we get about 20, 30 micrometers by measuring pH and oxygen actually. So what actually is driving the 400 micrometers you have shown around the root zone? So why is it 400 and why is it not 800 and why is it not 20? So is it a limitation of the availability of the phosphorus that drives the gradient, or is it on the other side, is it the activity of the plant root to provide the enzymes, or is it both, or how could you explain that?
So from the roots, we have some extensions, the same as from the hyphens. We have some extension of the various processes. And I think this is a group of the processes going in this direction, a group of the processes going in this direction. And the extension is very strongly limited how fast microorganisms will utilize this carbon, because there is not a lot of diffusion. The diffusion takes just some dozens of micrometers. It's not 2-5 millimeters. These are larger substances. For example, if we think about enzymes, they're huge in size. They don't diffuse in soil at all. And in my view, the diffusion is mainly connected with the diffusion of initial low molecular organic substances, which microorganisms use and then produce the enzymes. So it's not the diffusion of enzyme which was released by roots for half millimeter, for example, or 1 millimeter, but this is diffusion of, for example, sugars or amino acids, carboxylic acids to some distance until microorganisms completely trap it. And this is one reason, trapping by microorganisms. Another reason, for example, for amino acids, it could be sorption on clay minerals. It could be some other reasons too. So we have processes which go in this direction and go in this direction. And I think this is some kind of the stationarity because of the opposite effects of opposite processes. And coming back to your question, I fully agree that although hyposphere is local, very interesting hotspots, I can just not present it because, to regret, we have not any. So we are not so exact like you.
No comment on that.
Thank you. So it's interesting in the hotspots, but one of the limitations that everybody has with using carbon-14 labeling is that it treats all carbon the same. That is, your sugars, your amino acids, every carbon-containing compound gives you C14 in your hotspot. But biology doesn't treat those the same. Bacteria don't treat them the same. So that when you use C14 as your tracer, you don't have a good picture of exactly what the signals are that are going out into the soil and what exactly. I mean, I don't think plants are stupid, so they probably know what they want around their roots, and they probably don't. Not every hotspot's going to be the same.
Surely,
yeah,
they are different. I did not get completely. Do you mean fractionation of isotopes? Or
No.
No.
So when you label with C14,
yes,
it's a general label. We do this with C13 mostly.
Yes, we do.
But when you label generally with carbon, you label everything. So if you pick up the hotspot, what you'd really like to do would be a nice metabolomics experiment where you look at what made that hotspot a hotspot, right?
I'm sorry, I still did not completely got. Surely, if we label with carbon, we label all organic substances.
Right.
But we can do the same with phosphorus. We can do the same, for example, with nitrogen. Then we label only phosphorus substances or nitrogen-containing substances.
Yeah, but there's no radioisotope of nitrogen. So.
For nitrogen, not, but for phosphorus, there are two.
Right.
And very useful.
Right. I have a lot of people who come to my lab to do that experiment, but I don't do it.
Okay.
But no, I just meant that for the bacteria are going to respond differently to different substrates.
Sure.
Right?
This is maybe related to what I answered before. We can investigate factors, but I think is not correct generally to speak about various things are different. We need to define which things we would like to investigate and then define factors which, in our view, should affect this property. And then we do experiment and decide yes or no. So if you tell me very specifically, I will try to answer specifically. Maybe I will have no answer. But to speak too generally is not the very right way, I think. So we cannot speak just various substances are labeled different. I fully agree with you. But let us make this specific. I will try to answer very specific.
Yeah, no, what I was suggesting is
surely
is to go the next step and to use something like carbon-13 label, where you can then go in and ask what actually is labeled.
Yes, we have studies with 13C labeling and investigation of incorporation into PLFA or by SIP, stable isotope probing, into incorporation into specific microbial groups, when many specific groups are labeled. This is possible to do too. Yes, thank you.
Okay, Jacob. Sorry. There is another question there? No. Oh. Okay. Cornelia first.
Hi, thank you, Jakob. Very interesting. My question is, your processes, hot moments and hot spots are strongly dependent on the water.
Yes.
If there is no water, then nothing happens. So, and
yes.
your experiments you carried out under ideal water conditions, I imagine. And so, I wonder if the water can be used as an indicator of hotspot occurrence.
So I fully agree. So the most experiment what we do, the water is not the factor. So in the most, but we have 2-3 experiments, especially for the release of carbon by roots and enzyme activities, for example, beta-glucosidase, depending on the water content. If the water content is less than optimal, not dry, but less than optimal, the activities decrease very strongly. Activities of the release of carbon, activities of enzymes. But surely water is very, very strong factor, and when we travel from Atacama to heat wave,
okay.
Yes, fully agree.
Thank you. Just the last question. My question is hanging later, but please go ahead.
Very short question. I don't need microphone.
Yes, you don't.
Jakob, thank you very much. I have one short question, which is, if I understood very well your presentation, that the size of the rhizosphere doesn't matter. Regardless, the plant, they have all the same mechanisms. That's what you said?
Not at first. Rhizosphere is.
But because you said this size.
No, I say this is stationary. The size is stationary. It means if you look on this picture, just by chance we have this picture, there are some properties which have distance about 2 millimeters size of the rhizosphere. There are some other properties which have between 2 and 4 millimeters, and there are some other properties which have larger distance. It means we cannot speak generally about size of the rhizosphere. We should define for which of the property And we worked mainly with grasses. Not only, for example, also ryegrass, but mainly, yes, crops. I cannot tell anything about the trees or about the shrubs. Probably there are a lot of differences, at least because of the size and the functions of the roots, surely. And age. Age makes a lot, you know.
I know.
Okay.
Thank you.
Thank you very much, Jacob.
Thank you.
Okay.
Okay, thanks a lot for those presentations. Just a quick message for you. We kindly ask to our international speakers if you could go to the registration place, which is in the main entrance over there, to sign our registration book for international visitors, please. And we have lunchtime. So we will meet again here at 2:45. Okay? Thank you.
invited speaker. Okay, my name is Milko Jorquera. I'm work professor at La Frontera University, and now they left the turn to my colleagues, Jorge Farias, please.
Thank you. First, I would like to give our apologies because Paul should be in the airport in the 4th p.m. Yeah, for the reason finish, and I got to go to the airport. Yeah. Okay. In the context of this presentation, I would like to say some topic about or characteristic about the Professor Long. Professor Long is a microbiology training, and over the last 25 years has become a world leader in combined innovation in bioinformatics, laboratory, and field study to discover new high-value biotech products from the nature, particularly from marine environments. Professor Long has raised more than 7 million pounds to fund his research. On the other hand, Professor Long is visiting professor in pharmaceutical at the University of Sao Paulo, Brazil, and other universities around the world. Is co-founder and chief scientific officer an independent academic lead startup company exploiting the compound produced by the human microbiome to address specific health needs for the women. Okay, thank you, Paul, again. Please.
Great, thanks very much. Can everyone hear me okay? Is that okay? Yeah. Great. All right, so what I'm going to talk to you this afternoon about is some commercialization work that we've done where we've taken a marine natural product and turned it into a product that we actually now sell. And this is now in the area of photoprotection. And we sell our product as a cosmetic. So the route to market to a cosmetic is very different to what you heard yesterday when we had the veterinary medicine. It's a very different route to market. So what I'd like to do then this afternoon is to start off by giving you some general information about the effect of sunlight on human skin. I'd then like to talk about the work that we did in the marine environment that led us to a group of compounds known as the mycosporin-like amino acids, or MAAs for short. I'll then discuss some tissue culture work that we did, which led to intellectual property, which we could patent and then license to a company to produce our product, which is now available on the market. So to start off with then, just a brief introduction to solar radiation for those of you who are not familiar with it. So solar radiation basically can be divided into ultraviolet, visible or infrared regions of the spectrum, ranging from around 100 to 1,000 nanometers. The lower wavelengths have higher energy than the longer wavelengths. The ultraviolet region then is of high energy, and what reaches the Earth's surface is in the regions from around about 280 to 400 nanometers that are called the UVB and the UVA regions. When we think about how these regions affect the skin, basically there's a change in skin penetration. So the higher energy UVB radiation doesn't penetrate the skin as deeply as UVA. So just as an easy way to think about this in terms of photoprotection, what we say then is UVB, which is the higher energy, stands for burning of the skin, and UVA is for premature aging of the skin. So if we think about then what actually happens then to the cells, solar radiation then is a major hazard to all life forms. And we're only going to talk about UVR, okay, the ultraviolet radiation today, but increasingly cosmetic companies are really interested in visible light as well. So going out for lunch earlier today in the sunshine, that has an effect on your skin also, particularly causing pigmentation. So for the cosmetic industry, there's a big market in skin lightening. So the effect of visible light also is a potential market, also the world. So ultraviolet radiation then, the wavelength that it has, can be absorbed by macromolecules such as DNA that causes direct damage. And the direct damage to DNA is in the form of these CPDs, or cyclobutane pyrimidine dimers. The ultraviolet radiation can also be a photosensitizer, causing the formation of reactive oxygen species, or ROS, within the cell. And the ROS itself can also cause DNA damage indirectly by these 8-oxyguanidine type of lesions. You might think that if we have these lesions in DNA, it can cause genes not to be turned on. So that is the harmful effect. But actually, sometimes these mutations can occur in promoter regions, causing genes actually to be transcribed. And a really good example are the matrix remodeling metalloproteases, or the MMPs. So these genes are actually turned on and produce these MMP enzymes. And these have the effect of breaking down collagen. And visibly then, this is the wrinkles and the lines that we get in our faces if we have excessive exposure to sunlight. So we actually have genes turned on, causing photoaging. The reactive oxygen species can affect any macromolecule within the cell. For example, proteins causing oxidation of the proteins, and also lipids in the membrane causing lipid peroxidation. And if this occurs in immune cells that occur within the skin, this can lead to immunosuppression. I'm interested now in future work actually thinking about what happens to these macromolecules when they return back down to their ground state. When they return to their ground state, they'll be emitting low-level emissions of photons, which themselves may be causing some kind of damage within the cell. And it's not known what this damage is, or whether it's actually clinically even relevant or not, but I believe there might be an evolutionary significance to this. So I'm kind of interested in that, and I believe it might be a really good PhD project for anyone who might be interested. So, ultraviolet light then has an effect on human skin, and it can be severe initially, causing sunburn, and can have long-term effects such as skin cancer. So much so, the World Health Organization has this solar index, and basically, depending on the amount of UV radiation that you're exposed to, you have preventative measures that you can take in terms of sun avoidance. And it's ranked then from 0, where no protection is required, up to +11 for extra protection. And of course, it's color-coded to make it easy for consumers to understand. I hope that this is correct. I had to rely on someone else to write this for me, so I hope it's not rude or anything. Where do you think Chile sits on this scale? Down here or up here? Very much towards the right, do you think? Okay. So let's have a look at a map taken by NASA satellite then, looking at the UV radiation hitting the Earth's surface on Latin America. And I think we're here. Yeah? Okay. So you can see there, extra protection required, even on a day like today. Okay. So, for around about 80 years or so, synthetic sunscreens have been available. And cosmetic drug formulations of sunscreens either contain inorganic molecules, and these basically act something similar to, like, very small mirrors on your skin, reflecting the ultraviolet radiation. And there are also organic molecules, and these mainly absorb UV radiation, releasing it or dissipating it as heat. And here's just an example then of these organic filters. So a few years ago, the BBC made a program about the work we were doing. And this guy, he's quite a famous presenter in the UK. So he basically applied some of these organic filters to his face, and then we took a UV absorbing camera, and you can see the effect here of it absorbing the UV radiation. So you can see how effective this commercial sunscreen was. We just went to the local pharmacy and just bought something for him to use. So it's quite effective. So these synthetic filters have been around for a very long time. You know, some of us who are old enough in the auditorium may have been using them for 40, 50, or maybe 60 years plus, thinking that everything is great because they're good for us. You know, we're avoiding the harmful effects of UV, but this may not be the case now. Certainly, there are environmental concerns with these synthetic sunscreens. So there's lots of literature now available showing how studies have reported that the filters could cause hormonal changes in fish. There's something like 150,000 tons of synthetic sunscreen released into the oceans each year, and that comes from tourists. Yeah, so people just using it and then just going into the ocean, releasing all of these organic filters. And of course, you know, these don't break down very easily. The idea of a sun filter is you put it on your skin and it stays there, yeah, or washes off, but it doesn't break down in the sunlight. You know, it wouldn't be a very good filter if it did that. So it hangs around in the environment for a really long time. So hormonal changes as well. Because it hangs around in the environment, there's a lot of evidence for bioaccumulation now up the food chain and found in birds and marine mammals, so the cetaceans like whales and dolphins. And certainly the inorganic filters can fuel algal growth. And we've heard yesterday, I think, about algal blooms as well. So concerning is this, certain nations around the world and states in the United States have passed legislation now banning some of these synthetic sun filters, such as the oxybenzones. So last year, after many, many years of lobbying, it's now illegal to sell oxybenzone containing sun filters in the state of Hawaii. And legislation is now passing through California and Florida for the same effect. It's also illegal to import and sell oxybenzone containing sun filters in the Dutch Antilles and British Virgin Islands as well. And some of the Pacific Islands are also thinking about this. And you can see here the damaging effect that's been attributed then to these filters, basically killing coral, which is really important for tourism industry. The European Union is also really concerned about this in terms of human health as well. So in the European Union, we have something called CORAP, which is a Community Rolling Action Plan. And the idea of this committee then is basically to look at any chemical that is licensed for use in the European Union from a health perspective. And there are something like 28 synthetic sunscreens approved for use in the European Union. About 16 or so are used a lot, used frequently. And 8 of these now are on this CORAP list of concern for further toxicological studies because of their potential harmful effects either to the environment or directly to human health. So we have these oxybenzene type compounds here, but also interestingly, we have these inorganic filters as well. Okay, and these are regarded as cheap filters. Okay, so these are the ones which are used really widely. And what I found interestingly is sort of like the zinc oxide then is of concern because of nanoforms that may be produced, so nanoparticles. And these are thought to encourage formation of reactive oxygen species, which if you remember from my earlier slide, can have devastating effects within the cell. So I basically work at King's College in London, and if you don't know where King's College is, it's right in the centre of the city. You can't get more central than King's College. So if you want to come to King's, it's where it all happens as well. Yeah? Theatre, nightlife, everything. But I'm interested in marine biology, and a bit of advice that my PhD supervisor gave me, which is probably the best bit of careers advice I've ever had, was he said to me, you're starting your academic career, don't work in the area I work in because you won't be competitive. And he's absolutely right. How would I be able to be competitive for grant income against my PhD supervisor? He said to me, find yourself your own scientific research niche, which is not so easy, but he was absolutely right. And at that time, marine biology was still very classical in its approaches to the types of techniques it was using. Molecular biology hadn't really hit the marine biology world. So I thought with my background, this was an area that I could get involved in, my niche. But marine biology in the centre of London, where are we going to do it? In the River Thames? So I needed to find, fieldwork area. And for that then, I chose the Great Barrier Reef in Australia. So the Great Barrier Reef here is about 3,000 kilometres in length along the western coast of Australia. And Townsville, this city here, is where the Australian Institute for Marine Biology is based. And they have direct access then to the Great Barrier Reef, and they have fantastic facilities, including ocean-going vessels, which are equipped with laboratories, accommodation, even their own cook, their own chef. So you can stay out on the reef for weeks at a time doing field work. And I was working out on the barrier reef doing something completely different to sunscreen type of work. In fact, I hadn't even thought about it. But one afternoon, I was standing on the deck of the ship, and I was just looking out at the horizon, and it was a sunny day, and I felt my skin burning. And at that time, it was a low tide, so the corals in the lagoon were also out of the water. So I thought to myself, look, if I'm burning, why aren't these coral animals burning as well? Why don't corals get sunburnt like I was getting? And the answer to that was, people thought they made their own sunscreens. Okay? That's what people thought. What are these sunscreens? So these cyclohexamines, cyclohexanone rings with substituted amines or amine alcohols. And they're commonly produced by marine algae, seaweeds, and also found in tissues of certain marine animals like corals, which accumulate them through diet. Here's some typical MAAs, which are found in coral algal symbiosis. And what you can see here are the UV maxima. They tend to be in the UVB region, perhaps in the UVA as well. And for this reason, people had assumed that they were photoprotective, simply based on the UV maxima. And also from some fieldwork data, where people had dived down at different depths, and you find that you get a decrease in concentration of these compounds the deeper down you go. So obviously closer to the surface, more exposure to the UV. So really sort of like anecdotal type of evidence. So I thought, well, what I could do is use molecular biology, cell biology type of techniques to try and prove whether these MAA compounds are photoprotective or not. So what we then did was just look at the diversity of these MAA compounds in animals, because we're animals too. And what we found was that they're widely accumulated, presumably by diet, in different invertebrates, in fish, in the roe, and this is the ocular lens of a dolphin. And what we found was that we always get mixtures of these MAAs. They never seem to occur individually on their own. They're always there as a mixture. And it's usually the same mixture in the same kind of ratios. And that got me really interested in why. Evolutionary, why would you have a mixture of these compounds? We don't know the answer to that. We can just speculate. And again, I think that's quite a good PhD project for anyone who might be interested. However, the compound that you always find in large amounts is called palathine. So because it was there in large amounts, we'd have access to it. That's the one we decided to study. Okay, that became our model MAA. I want to digress just slightly because I was looking at a few of the posters yesterday, and a lot of people are interested in using natural sources as ingredients for different types of things. So what I thought I would do is just show you this slide, because there's always a sustainable supply issue when you're working with a natural product. If you want to commercialize a natural product, you have to have enough to sustain a global market. Okay? So sustainable supply is a huge issue. So what usually happens is that these natural compounds are present in really low amounts. So you have to collect a lot of biomass to get a lot of compound. So there are environmental issues for large-scale harvesting. And of course, if you harvest too much, then we have this issue then maybe of species extinction as well. Structural complexity of some of these natural compounds usually prohibits chemical synthesis, certainly economical synthesis on industrial scale. These MAA compounds look quite simple, but believe me, their stereochemistry is really challenging, and we've yet to find a starter material that is commercially viable for us to use. So very difficult. What we could do is some kind of tissue culture or aquaculture as well. Or there's always heterologous expression, so we can take the genes that make the compounds and express them in a heterologous host. And for the MAAs, this is a real possibility when the source is cyanobacteria. But going back then to our MA story, the palathine. Our source of the palathine was the edible Buddha's ear seaweed. And the reason why we chose this was, first of all, it's farmed commercially in a sustainable way. So it's seaweed grown on a rope, essentially. And it's edible. It's used in cuisine in Southeast Asia. So it's not likely to be toxic to humans also. So what I'm going to show you now are some of the in vitro studies that we did using purified palathine to try and show whether it was photoprotective or not. So I'll start off looking at its solar range and photostability. Remember, photostability is really important if you want to have a commercially viable sunscreen. Some biological assays, such as cell survival, DNA damage, direct or oxidative, induction of these reactive oxygen species, some gene expression, and assays for antioxidant effect. When we're doing tissue culture and we want to have a source of radiation, there's no point just going out, taking the tissue culture flask out into the sunshine. We need to have something that's a bit more controlled than that. So we have something called solar simulated radiation, or SSR for short. And this basically is a high intensity bulb used clinically to treat certain types of skin conditions. And if we look at a typical solar spectrum, you can see the wavelengths of light that we can get just from exposure of solar radiation. And this was taken at noon on Ipanema Beach at Rio. If we take now a look at the solar stimulated spectrum, it marries quite nicely with what we would expect to get from normal exposure. So our solar simulated source is a good source for our experiments. Interestingly enough though, the intensity of the irradiance is much higher with the SSR source. It's equivalent to being in the Arizona sun at midday. So it gives you an idea of the intensity of the irradiation. So it means we don't have to expose the cells for too long. If we have a look at the absorption spectra of the palathine, you can see that the absorption maxima is around about 320 nanometers. So that is in the UVB region. If we compare it to one of these oxybenzones commercially available, you can see that the oxybenzene has a much broader spectrum. That's why it's sold commercially as a sun filter. It gives us broad spectrum protection. Interestingly enough, though, that we do have this tail in the absorption spectrum, which goes into the higher wavelengths of UVB. So the stronger wavelengths of UVB. And that's important to hold in the mind for a little bit later. If we think about the photostability then, what we did is we exposed the palathine then to different intensities of the radiation. And this is measured in what's called SEDs, which is the standard erythromemal dose. So this is how long it takes you to burn. And this is standardized as burning after 5 minutes, this type of dosing. So it's a standard way that the cosmetic industry uses to determine the efficacy of filters. 15 SEDs is probably what you would get if you went out into Ipanema Beach all day. Okay? That gives you an idea. Can you imagine what you would look like after a whole day on Ipanema Beach without any sunscreen? Okay? Gives you some idea of the strength we're talking about here. And what you can see here from this graph is that the palathine seems to be really, really stable, even with 50 SEDs. And here's just a few numbers here. 3.6% degradation. So it's really stable. Cell viability. We don't want the palathine to kill human cells. That would be very counterintuitive. Although we probably were in a good space because we know that the seaweed is eaten. So what we did was we took primary human keratinocytes. So not cell lines, okay? These are primary cells taken from human volunteers and grown in tissue culture. So the keratinocytes look like keratinocytes, not like Hickok cells, which were keratinocytes, but don't look anything like them now, okay? So 100% viability in our control, which is what you would expect, and without any exposure to radiation, but with 10% palathine, also 100% viability. So we expected this result. Why 10%? 10% is the maximum concentration of an organic you can add to a formulation under European legislation. So that's the maximum we would be able to add. When we expose the cells then to radiation, we get a significant drop then in our controls in terms of their viability. No surprise there. But when we add different concentrations of the palathine, we maintain our viability. So there's a photoprotective response here. The palathine is protecting the viability of the cells. Anyone a pharmacologist here? Anyone does pharmacology? Yeah? Okay. You notice here we've got different concentrations of the palathine, yet the viability remains the same. I would expect a dose response, but we don't see a dose response here, and I don't know why. Okay, no idea. Maybe it doesn't matter. You know, maybe it's an all or nothing thing. The important thing, though, is viability is not affected. Now, if we start thinking about DNA damage now, we can use different enzymes to look for different DNA lesions, and these are the alkaline label sites, the CPD dimers, and then indirect damage through ROS, the 8-oxyguanine lesions. And we used our solar simulated radiation, so that's across the whole set of wavelengths, and then we put a filter in, so we would just be looking at the really damaging UVA region. So here's our control then, no exposure, without the palathine, and we get 100% DNA damage, is what you would expect, across all of the different lesions. No surprise there, is what we're looking for. When we then add the lowest concentration of the palathine that we've been testing, what we see is a significant decrease in the amount of DNA damage across all the different types of lesions, including the ones that are formed by reactive oxygen species, so indirect DNA damage. Why is that important? Well, what happens to you after you've been out in the sun and then come back in the evening? What happens to your skin? It burns. It burns after the event, okay? That's the effect of the reactive oxygen species carrying out what is called dark chemistry reactions. So what we're seeing here is a reduction in dark chemistry reactions, which was a real surprise to us. And remember, patent lawyers loved that phrase, was it a surprise? And this was. And even more surprising, when we just looked at UVA region, we also got a significant reduction. Remember what the absorption maximum was for palathine? It was in that UVB region, but a little bit into the UVA. So we're getting significant protection then against DNA damage caused by UVA radiation just at that high intensity wavelengths, not across the whole spectrum of UVA. Again, that was a big surprise for us. In other words, do we really have to have a broad spectrum sun filter? The answer is no, probably. If we look at gene expression, I won't go into this in any detail, but essentially we were looking at inflammatory markers and also markers for immunosuppression. And here are those MMPs as well. Irradiated controls, we see damaging gene expression, but also then we get a significant reduction in this damaging gene expression also. So it's exactly what we would have wanted. Induction of ROS, reactive oxygen species. Again, in our controls, when we just expose the cells then with no radiation, just grow them normally, you do get a baseline of reactive oxygen species forming. That's due to normal cell metabolism. Okay? We carry out oxidative phosphorylation, which by definition generates reactive oxygen species. Okay? That's part of our normal metabolism, so that's what we see here. When we expose the cells then to radiation, we get a significant increase in the amount of ROS formed. Again, exactly what you would expect. But then when we add the palathine, we see a significant reduction in the amount of reactive oxygen species that are forming. So the palathine then not only has optical properties that are important, but it also seems to have this reactive oxygen species quenching properties as well. And again, what we see here is that if we add the palathine after exposure, we can also significantly decrease the amount of ROS that is forming. So these dark reactions, in other words. So how may the palathine be working then as an antioxidant? There are various mechanisms how this could happen. One is by radical quenching by hydrogen atom transfer. And what we can see here when we looked at 3 different mycosporines, you can see that there is a response then to different concentrations. So they're definitely quenching these hydrogen atom by hydrogen atom transfer, quenching the free radicals. Our control is ascorbic acid, so that is vitamin C. That is the industry standard, okay? It's to add vitamin C to your sunscreens. Okay, so if you buy sunscreen, look at the ingredients, there's always ascorbic acid added to it. That's because sun filters commercially, these synthetic ones, have no ROS or anti-ROS activity. You have to add it to the formulation. So again, this was a bonus for our compound. And Trolox is just an analog of ascorbic acid. We also looked for electron transfer as well, and there was no radical quenching here. So the mechanism there was likely by hydrogen atom transfer. So the palathine itself then chemically acts as an antioxidant. But if we think about what our cells do to try and quench these reactive oxygen species, our cells produce enzymes and other proteins that deal with the damaging effect of free radicals. Many of these proteins and enzymes are under the transcriptional activation of an activator known as NRF2. So it's a master regulator, and not surprisingly, it's a very druggable target for the pharmaceutical industry in any disease that has ROS implicated in its etiology. Alzheimer's, Parkinson's, cancer, old age, anything like that, metabolic diseases. And NRF2 is constitutively expressed in the cell all the time with a regulatory protein called KEAP1. So what happens then is that NRF2 binds to KEAP1. It's its regulator. You don't want to have your proteins turned on all the time if there's no need for them to be turned on. What happens then is that the NRF2 is targeted for ubiquitination, and that's how the cycling works. What happens though, that in the presence of sunlight or UV radiation, ROS is formed, and the ROS then can basically react chemically with any residues that are chemically susceptible. So a really good example then will be thiol groups in the protein structure. And this breaks down the interaction between KEAP1 and NRF2. So Any compound then that breaks down this interaction really is the drug that pharmaceutical industries is looking for, okay? So what happens then is NRF2 becomes freely available, it migrates back into the nucleus and becomes bound to an antioxidant response element. And this then leads to high expression then of these proteins involved in cell defense, okay? So at that time, I had a PhD student who was doing computer modeling of KEAP1 NRF2, and he built a really good model. And I said, well, why don't you try these mycosporins just to see whether they might have an inhibitory effect. So what we did was we took the KEAP1 protein and the specific part of the protein, this couch repeat, which is essential for the binding of NRF2. And as a control, we have this peptide sequence that has 100% homology to that sequence. So in our assay, which is a fluorescence polarization assay, you get 100% then inhibition with the control, what you would expect, because it binds directly, 100% homology. What was really surprising was when we looked at a couple of these MAA compounds, these also significantly bound to the KEAP1 couch domain. This was a real surprise to us, because this had never been shown for a natural product before. All these natural products here, okay, have been described in the literature as inhibitors of KEAP1 NRF2 binding, but they're non-specific. They chemically react with thiol residues in KEAP1, causing the complex to fall apart. So they're non-specific. This was the first example then of a natural product, group of natural products, that are specific competitive inhibitors of NRF2 KEAP1 binding. A big surprise for us. So, would that result in an upregulation of any of these cytoprotective proteins? An example that we use is glutathione, or this protein involved in glutathione biosynthesis. And just to remind you then, this is the KEAP1 protein and how NRF2 binds to it. And the idea then is that the mycosporins will competitively compete for this binding site, releasing the NRF2. And you can see here from our controls, where we don't have the presence of the ultraviolet radiation, there's no competitive inhibition because there's no need for the genes to be turned on. But in the presence of the UV light, we get an increase in NRF2, which we would expect by the non-specific binding. And also here, this is sulforaphane, which remember, this is the chemical that indirectly is reactive to any thiol residues, causing disruption of the complex. But also here, what we see is significant increases in expression of this protein. So we have evidence then at the cellular level that we have competitive inhibition, of KEAP1 NRF2 binding. And this is the first example then of a natural product that does this. So what are the requirements then for an effective sunscreen or antioxidant sunscreen? Well, we have to have significant absorption, which we showed. Not only does the compound give us protective effects in the UVB region, but also in that tail of absorption in the high energy UVA region as well. We have sufficient photostability, which is essential. It's non-toxic. It's highly soluble. It's colorless. Why does that matter? Well, I don't know, to some people maybe not. Maybe you want to put color on your skin or in your hair, maybe. And it also prevents UV-induced damage as well. So, not only do we have optical properties, we also have anti-ROS properties as well in a single compound. This gave us patent position. So we filed our patent and we had our priority date in 2015. We filed for the PCT filing then in 2016 and published. And then the patent was granted by European Patent Agency in 2020. So only 4 years then after the filing date, we got the patent granted without any comments coming or challenges from anyone else. So, you know, we were really pleased with this. You know, it always surprises me when, you know, some academics, for example, might turn around and say, I've got 20 patents. And I say, well, how many of them were granted? Oh, none. We don't have a patent then, do you? Yeah? It's something to think about. Okay? Something to think about. So, this is all done in the university, and I work for the university. So although I'm an inventor, I don't own the intellectual property. Yeah? A lot of academics think that because they're inventors, they own it, but they don't. Your employer does, right? So although the university might pay for the initial filing of the patents, it wants to get its money back. Not only for the patent filing, but also for all the experimentation work that was done as well. Everything it wants back. And then what it wants is to make a profit too. So it doesn't want to sell the patent immediately, it wants to license it to a commercial partner. So licensing is what's really important. So the licensing then will have a fee that will recoup all of the investment, but also will have a reach-through profit-sharing agreement where the university then will get a certain percentage based on sales or promises of sales. There's a difference as well, okay? So the company that we chose was called Ethic, and notice the way that Ethic is spelt here, okay? This is an ethical company, and we quite like the idea. And I mean, everything that they use is biodegradable and all that kind of stuff. So, you know, the box is made out of rice paper, for example, and all this biodegradable plastics and things like that, okay? And what we decided to do was, rather than try and get regulatory approval for these microsporins as a sun filter, what we decided to do was to sell as a cosmetic, as an antioxidant. Why did we do that? Well, it was really explained well yesterday. The clinical trial types of things that you have to go through and the costs of clinical trials. For a sun filter, you'd have to make a specific claim that it prevents UV damage. Okay, it's a specific clinical claim. So you have to go through all these clinical trials. It can cost for a sun filter around about 10 million US dollars and takes around about 10 years. So we decided to go for a cosmetic because you don't have to do any human trials, okay? But you mustn't make any health claim at all. So look at what we did here. You know, one long-lasting triple solution ingredient, you know, reduces the signs of light-induced damage. Yeah? So very broad kind of claims, nothing too specific. Okay? Yeah, So this is like, you know, what the commercial people said to me, you know, it's marketing beats tech is the phrase that they use. Okay. Anyone want to buy some? It's available online. 270 euro for 50 gram. It's cheap. Yeah, cheap. All right, so just to summarize then. So UV exposure initiates a lot of molecular changes in the skin that can lead to cancer and photoaging in the long term. Sunscreens can prevent molecular damage, but there are environmental health and now perception concerns as well. These microsporins are natural, biocompatible compounds that have been extremely effective at reducing induced molecular damage in vitro. And the MAAs have these antioxidant properties, and this is an advantage over the synthetic sunscreens because they don't have this property at all. So as I said, if you look at your sunscreen, it always has vitamin C added to it. Right? Vitamin C actually breaks down really quickly when it's exposed to sunlight, so it doesn't really work anyway. Right? So I think we have a better balance with nature. For the first time, we've shown that we actually have in a single compound optical properties and also antioxidant properties by two different mechanisms, direct chemical quenching, but also induction of the cell's normal defenses also. And that was a first for us too. The supply issue is always going to be an issue with a natural product, and that's what keeps the price high. So just some acknowledgements then. Anthony Young, he's a dermatologist at King's I've worked closely with. 2 PhD students who've gone on now to successful careers themselves. Vanessa Geraldes from University of Sao Paulo, Butantan. Again, she's now postdoc at Butantan. And another collaborator, Anani Pinto from USB Piracicaba. And our funders. And thank you very much for your attention.
Thank you very much, Paul. It's open for the, round of the question or comment. Please.
Hi, Mark. Congratulations for the exposition. A question: how could you patent this molecule if this molecule is in the nature?
Yeah.
How you did it?
Okay, right. So this is what's happened recently in the last 10 years or so. You can no longer patent a natural product anymore if you're patenting the natural product for something that it does naturally in the environment. But okay, if we were going to patent it as a sun filter, I would agree with you. Probably the examiner would have an issue with that. But actually, where do you actually file the patent? If you filed it in the United States, you would definitely have a problem. Okay? But in other parts of the world, they're a bit more lenient about the natural products. And what we really went for was for the antioxidant properties, which were never described. So that's the reason. So the antioxidant activity is something that we're saying doesn't occur naturally in nature. Probably does, all right? But really it's the optical properties we think that the organisms that produce these compounds are using them for. And that's probably why they make mixtures of these.
Okay. And some advice from you about the lawyers and the lawyers' company we need to choose to make things like this, because what you did is very, very tricky.
Yeah. Okay, it's so tricky, it took 20 years to do this, all right? Most of the time, for that very reason, for commercial reasons, because every company we went to, you know, like Johnson & Johnson and all these big companies, you know, that make sun filters, they said, look, this is no good to us unless you can produce 1 kilogram for less than $50 US. Now you show me a synthesis that you can do for that in bulk for an organic. It's really difficult. Profit margins on sun filters is really, really small. But in the last 20 years, with global warming and the effects of the planet and all that kind of stuff, consumers are changing their choices. So now it's more economically viable to take something that wasn't very viable 20 years ago. So it's taken us a long time to get the funding in place to do all of this. So knocking on lots of doors, like I think you were describing yesterday, knocked on lots of doors, got a lot of rejections, but you just have to keep on going. In terms of picking a lawyer, well, it doesn't really matter, does it? As long as they're a good patent lawyer, they all charge a huge amount of money, all right? But the thing is, if you're working with a university, the university, through their commercial exploitation activities, should have a budget for it. The problem there, of course, is you're in competition with everybody else. There's only a limited amount of money they have to support patent activities. So usually universities have a portfolio which they support. So in my case, it's small molecule therapeutics and diagnostics, which this did not fall into. So I was very unpopular, you know, really unpopular. Does that answer the question?
Yes, great. Great.
Yeah. So keep on going is what I would say.
Yes, I can, I can go on.
Yeah, no. I mean, in what you do, keep going. You know, don't. If the door slams, just try and open it somewhere else. And because we kept on going for 20 years doing this, the market changed, and the market has driven what we've been able to do.
Great. And you, like a researcher, and in your position like an entrepreneur, what competence, what skills you need to do to pass from a researcher to an entrepreneur?
Trial and error. There's a lot of errors. I've learned so much by doing this. You know, I find a lot of PhD students are really keen on sort of like becoming, commercializing their research and everything like that. And my initial reaction is, don't do it, all right? Because it is really hard. Because if I wanted to be a businessman, I would have gone to business school, all right? It is really, really difficult. I always say that the moral compass of a businessman points in a very different direction to mine as a scientist. You know, the way they behave is very different to the way, scientists behave. They're very cutthroat increasingly cutthroat. Some of their behaviours that I've seen, I would never behave like that, but it's very normal to them because it's a competitive market for them. You know, it's normal behaviour. So enter into this, you know, with your eyes open and with a very thick skin. All right, that's what I would say. But hopefully the university will have people with experience to try and help you. Sometimes it's a hindrance, you know, but you basically get as much advice as you can from as many people who have done this already. So scientists who have successfully done this are the best people to get advice from, because they'll tell you, you know, where they fell down, things like that. But the biggest hurdle, I would say, is getting that initial investment. Because what investors will always say is, when will I get my money back? And is the patent protected? That's what they always say.
So we should link the business and science together.
Yeah, of course. Yeah, because what the business people will say is, even though they don't necessarily understand it, they'll say is, how comes this is different to something else that's on the market? And, you know, can it be done, you know, in the Far East? Much easier. Can it be copied, in other words, as well? That's really what they want to know. They want their money back, and they want it back by, you know, 2,000%. Okay? We're not talking like, you know, markup in the supermarket.
10 times.
Yeah, they want 10 times.
Yeah, well, one important part of this story is because many industry doesn't sell product.
Yeah.
It's concept.
It's concept, totally.
Right?
Totally concept.
So that's the difference.
Totally. So, you know, Max Factor, for example, you know, they change their product line, usually 12 to 24 months, because they've been copied. So the mascara and lipstick and everything will change slightly in its shade or name because it's a concept change. People think that I'm getting something new. Completely, completely. Yeah. And it's all to do with, you know, shelf space and everything. It's a very different thing. So in my business, for example, I don't handle anything to do with the business side of things. I mean, my nerves just can't take it. You know? I stick to, when people say to me, what do you do for the business? I say I'm the intellectual property. Yeah? So I just say, this is what I do. The commercial people do the commercial thing. Play to your strengths. Don't try and do everything. You know? Don't try.
There is time for the one question, the last question or comment. Okay, this.
Do you think we as scientists might change this behavior from the businesses? Because we have to change our business model. What they tell us that they are old-fashioned. It was developed in the 50s, and now with all the environmental problems and everything, we can't go on like this. So how can we make them change?
Yeah, again, you know, we had this argument over 20 years with the industry, and they're not interested. It's the bottom line on the Excel spreadsheet that counts. That's really as it is. So no matter how much lobbying you do, trying to convince them that it's environmentally friendly, on a personal level, they may agree with you, but in the boardroom, it's not. It really isn't like that.
But how can they, when they agree on the personal level, they can change something?
Yeah, it doesn't work like that. No, it doesn't work like that. It's only when you have, you know, a big catastrophe, you know, that will, you know, it's about building momentum. So, you know, it took something like 5 or 6 years for the law in Hawaii to come into effect. People were lobbying for years for this to happen, and it just took such a long time. So yeah, it's all about lobbying, who you speak to, who you know, and in business, of course, friends are friends, but keep your enemies closer. You know? Yeah. It's who you know will open the doors for you in business, for sure.
But it's very tough to be a businessman, and very tough to maintain a business alive.
Yes.
And when you are a businessman, in the Sunday evening, you have butterflies in your stomach because you need to pay salaries,
Absolutely.
you need to pay rents, and
So again, what I would say to that as well is, you know, don't enter this lightheartedly. If you go into a startup or a spin out or whatever you want to call it, be prepared to work 24 hours a day, 7 days a week, for a long time before you even get a product approaching the market. You know, you could say goodbye to your family and your friends and things like that. it is 100% commitment, and there's no guarantee it's going to be successful. So really, you know, it's very glamorous having the idea, oh, you know, I'm a co-founder and, you know, chief scientific officer of my own spin-out. You know, it sounds lovely, but the reality is very different, very different. Other, you know, if it was easy, people in business wouldn't have to do it full-time, would they? So we're trying to have two hats now, being a research academic and, you know, on the business side too. And what I found is you can't do both. You have to pick one or the other eventually.
Okay, thank you very much, Paul, for your interesting
Thanks.
presentation. Now I want to go to the airport.
Okay, thank you. Okay, our next speaker is Dr. Fumito Maruyama from Hiroshima University, Japan. Dr. Maruyama is graduated from Osaka University with formation in field of microbial ecology and environmental microbiology, and currently he is acting as full professor in section of microbial genomics and ecology in the Hiroshima University. He also acts like an ad honorem professor in La Frontera University. His research activity of Dr. Maruyama are mainly focused in elucidate the role of microorganisms and algal bloom and their interaction, and to decipher the genomics and metagenomic diversity and dynamics of microorganisms in aquatic and air ecosystem and pristine ecosystem affected by climate and anthropogenic induced change. The third one also is research focus in the characterization of genetic and biochemical mechanisms involved in the microbial interaction and their living environment. And finally, to develop identification of pathogen, microbial pathogen, prevention and prediction of algal bloom for sustainable aquaculture around the world. Okay, this is a pleasure for me present because how do you know Dr. Maruyama is we obtained the doctorate degree in the same laboratory, and we had a very close collaboration in the last 20 years. Okay, the presentation is Dr. Maruyama is Chile-Japan International Collaboration open new research on environmental microbiology related to planetary health. Please, Dr. Maruyama, go ahead.
Okay, thank you for your kind and long introduction of myself. Maybe most my presentation is summarized by you now. So I think I don't need to present my talk today. So today's talk, as he introduced me, one of the main talk is related to aquatic microbe, which is focusing on harmful algal blooms. Maybe yesterday, Mirko already presented about the latest progress and achievement of the Chilean and Japanese collaboration. And so that's why today I will talk about a little bit different thing, because after the long time collaboration, around 20 years collaboration, we are now moving to new phases. Firstly, maybe from the pathogenic bacterial dynamics in the natural environment, and I always try to help the Mirko's research is related to plant growth promoting bacteria, but recently we were moving to new researches, which is a collaboration with younger scientists of our laboratories. For example, in the case of Mirko's laboratory, now Jackie is PI with co-PI with my assistant professor in my laboratory, Dr. Fujiyoshi. So firstly, I'd like to talk with what is my laboratory now. My laboratory now is focusing on the microbial genomics and ecology. As you see, this logo is designed by Dr. So Fujiyoshi. Almost all the philosophy in my laboratory is written in this logo. And then now my laboratory is also belonging to the Center for Planetary Health and Innovation Science of Hiroshima University now. The logo is here. This logo is also designed by Dr. So. This is Japanese hiragana, and this design is made by, font is also designed by Dr. So. And also, today's talk is mostly related to this logo. We say Chobe, but this project is related to microbe of the built environment. As you see, this logo is also designed by Dr. So. The project is related to microbe of the built environment, which the field is now hot topic in the world because under COVID condition, most of us always staying in our living environment, and microbe around us highly infect to our health. So maybe some of the people here don't know much about the planetary health. So firstly, I'd like to explain you what is the planetary health. So maybe it's a very simple concept. As you know, in the field of infectious diseases, public health is the most important philosophy, but after that, only a normal public health is not enough to consider the infectious diseases such as coronaviruses. In that case, global health is more important. But in the case of, for example, influenza viruses, we need to consider about animals in addition to humans. So one health concept is important. But recently, in addition to COVID, such as COVID and Zika viruses, to consider all of the world, our planet need to be considered. It means we need to consider human animals, and also we need to consider about biodiversity and natural environment. This is planetary health and now growing concept in the world. So as you know, maybe not so many people know much about Japan. This time, I come here from Hiroshima. It took 50 hours from Hiroshima to Pukkon. 2 days ago, I arrived here, but now jet lag is 12 hours, time lag is 12 hours. It means now is around 4 a.m. in Japan, so too sleepy for me now. And maybe some more people don't know much about Japan, so please see that this is a normal rush hour time in Tokyo. Maybe as you know, this is a train station, this is a normal scenery, and Tokyo has more than 1,000 stations, subway and a normal train station in Tokyo. Only in Tokyo has more than 1,000 stations. And every station looks like this, too much. But for example, in the case of other area, Japan has so many variety of scenery in Japan. So there are conventional area, traditional area, and there are so many shrine and temple, and maybe you know Japanese animation. When we go to the Akihabara in Tokyo, you can find this kind of so many animation building around the city. So there's big variety in Japan. And my Hiroshima University is located in suburban to rural area. In that case, you can see this kind of atomic bomb dome, and also some shrine, beautiful shrines, and there are so many local foods and local sake. Maybe you know sake, but in Japan there are so many different kinds of sake. Local, we can drink local sake only a limited area. And Hiroshima also famous in the local oysters. So if you been to Japan, you can find so many local food or local sake and local scenery you can find everywhere in Japan. So Japan is very, very diverse. And I think Mirko already explained about the current progress of our big Japanese and Chilean project. So that's why I will skip a little bit. But this time I come here to have a meeting and which is related to evaluation of long-term project. So that's why I come here for 3 weeks to have a evaluation by JST and JICA. That is the Japanese governmental entities. So in that case, we start from this big project 5 years ago, and as you see, there is a Japanese ambassador in Chile and Chilean ambassador in Tokyo, in Chile. So, and also there are so many rectors of universities. So it means that this project is very, very important international collaboration, as you see. This is the evidence. And also Chilean Foreign Affairs webpage shows this kind of news. So this is a very important, big, one of the biggest project, scientific project in Japan. So this is the final physical year of the project. So that's why now Mirko and I am so nervous about the evaluation. So I need your support to say, please say, this project is very good and nice. Again, to the some evaluators who will come maybe 1 week later, I think. And this time I hope to introduce my new research topics with Mirko also. So this topic is related to microbe of the built environment. So as I said, almost us now staying inside of the building of our time, so long time. And our building is highly tight in air, but all the days we are living in this kind of open air buildings. So this kind of building is developed maybe last 300 to 400 years in our long life histories. So it means our normal living environment now is very new environment. So it means for our life, building is a little bit totally different from natural environment. And I suppose this, our environment is one of the extreme environment for microbe and living life. So in our laboratory's topic, especially transmission of airborne microbe is very important. So this review is written from the 2017, more than 5 years ago, but this review is now citing so many people. And especially in the source of the microbe in the built environment is from ourselves and also come from the air. It means a microbe from ventilation systems. And also some specific bacteria can grow in our built environment. So especially this kind of source of microbe is especially from bathroom. And in the Japanese system, our normal house don't have bathroom with toilet. Usually we have a bathroom and toilet are separated. So it's a unique bathroom in Japan. So that's why not so many people try to search for what kind of microbe in our Japanese style environment. So in my laboratory, we try to clarify what kind of microbe in our bathroom environment. Firstly from water, from shower, and sometimes we took a sample from inside of shower head. And also we take a sample from all over Japan, but we don't need to take a sample from all over Japan because everywhere in Japan we can drink water because tap water everywhere in Japan is drinkable. So not so different in the community structure of tap water. But so for example, this is a result of a microbe from inside the shower head. So as you see, most bacteria is from the normal freshwater environment, but some bacteria is related to human commensal bacteria. But because this is the inside of shower head. Maybe in the case of water, the bacteria species is only related to water associated. So maybe in the case of inside shower head, our human can be the source of contamination to the inside of shower head. So this is the beta diversity. Bacterial community structure is similar or dissimilar. In the case of water, bacterial community is very similar compared to bacterial community inside the shower head. So as I said, all the tap water in Japan is drinkable and not so different between the locations. But in the case of shower head, the bacterial community is totally dissimilar, even if the distance of sampling point is very near or not. So as I said, many Japanese people drink the tap water as a normal water source. And as you see, there is not so many bacterial species are found. Maybe Legionella and Mycobacterium is a very famous waterborne pathogen in the tap water. But usually, I never found genus Legionella, for example. And this is a list of pathogenic genus, but I rarely found the important pathogenic species, and only some less virulent pathogen we can find. But this bacterial community structure is very different from EU and European waters. In the case of non-tuberculosis Mycobacterium case, US usually have around 15% of mycobacteria in the tap water. And in Europe case, around 6 to 7% of bacteria in tap water is Mycobacterium. But in the case of Japan, for example, the Mycobacteria are here, and its average is less than 1 point, 0.5, 0.4 in Japanese case. So totally Japanese tap water bacterial community is different from other world. So that's why now I'm doing the sampling every 3 months from the source of tap water. This is drinking water purification systems in a local government. So I hope I can find what is the reason of the difference between Japanese tap water and American tap waters. And also, if I check the bacterial community in tap water and biofilm in the shower head, I can find some human pathogenic genus is only inside the biofilms. So this plot is here you can see the alpha diversity, and this vertical line indicates the quantity and diversity of pathogenic microorganisms. So if we plot the alpha diversity and pathogenic bacterial diversity, I can find how safe is tap water is. So keep monitoring is very important, but if we use this kind of methodology, we can say if the water community is like this plot, maybe this water will be not normal and maybe dangerous. So this kind of plot is very useful to check the quality of water microorganisms. So currently, now we try to what kind of factor is affected to showerhead biofilm. Now I prepared 3 rooms without human and with human and human with biotope like this. Then I will clarify what kind of source will be the important for the showerhead biofilms. No one knows showerhead biofilm is from human or not. So I think this kind of experimental room with human will be the good experimental model to clarify what kind of human existence or presence or absence cause affect to the microbe in the shower head. And another topic is related to Japanese traditional house microbe. So maybe you can see this kind of room house. This is a traditional Japanese house. This roof is plant, and this floor is soil. And as you see, there is a bamboo for the ceiling. And this is a traditional Japanese space room for sleep. So I take several samples from inside and outside of this kind of traditional house. This kind of traditional house is still many in rural area, and many people still living in this kind of room. And we take a sample from air and wall and floors. And also, in addition, this is a collaboration with architecture scientists. It means transdisciplinary researches. So this kind of temperature and humidities are measured by architecture scientists. They usually take such kind of parameter every 10 minutes, for example. So after the investigation of DNA concentration and humidity, as you see, this kind of very, very high correlation is found between lowest annual relative in humidity with biomass of microbe in the area. So this is very rare, and this is the first findings of the correlation between this kind of lowest annual relative humidity with microbial biomass. So usually if we do this kind of research by ourselves, we usually use only average relative humidity. But if we use the lowest annual relative humidity, we could find this kind of very too high correlation. And this kind of traditional Japanese house has a very, very unique bacterial community structure. There is a very interesting point is in the case of earth floor is a soil floor, as I said. This floor is usually made from just soil with sea water. So that's why, but I don't know the exact reason, but more than 70% of bacteria is consisted from archaea, Halococcus. Because Halococcus is a very specifically exist in the high salinity lake, but in the case of earth floor in Japan, we never found this kind of high concentration of Halococcus without this earth floors. If we make a earth floor once appropriately, earth floor can continue more than 200 years without any arrangement or cleaning. So it's like earth floor will be one of the extreme environment. And also, in the case of tap water, as you see here, around 20% is a candidatus nitrosophila, because around this region or in the rural area in Japan, still many people drink the underground water. So that's why many area in Japan has many source of underground water. It means that there is a rich source of new bacteria. So no one care much about the bacteria in the underground water, but I believe Japanese underground water is very safe for drinking, but in addition to source good bioprospecting source of new bacteria for biotechnology, I believe so. So that maybe, I hope this kind of new bacteria can affect to the taste of sake, local sake. I believe so. And so this is a summary, but what I want to say is that our Japanese traditional rural area house is totally different from normal modern houses. Because inside and outside of the bacterial community is usually, many people say, totally different. But in the case of traditional Japanese house, inside and outside bacterial community is almost same and never distinguished between inside and outside. So I think it means Japanese traditional house is like outside. We can live inside with outside environment in the view of this. So Japanese rural area need to go there by so long time to reach there sometimes. So we bring this kind of material, suitcase laboratory, to analyze the bacteria at the on-site. So this is created for the this Chilean and Japanese international collaboration. It means SATREPS and project, but this device is also used for our new collaboration. And this is another devices. This can monitor the aerosol, bioaerosol bacteria on real time. So this is just an example of a measurement. Maybe no one never see this kind of data. Above one is PM, particulate matter, in a room and when opens a window, and outside the particulate matter. And this plot indicates living bacterial microorganisms. So as you see, in Japanese cases, if we open the window, microbe will increase, and outside is more. But usually, European people say opposite. Closed room has a more bacteria, and when we open the window, nothing changes, someone says, and outside the lower number of bacteria. But in Japanese cases, it's the opposite. But about the chemical, physical parameters, the tendency is the same as the European cases. Of course, closed room, now here, for example, higher CO2 concentration compared to outside. So this physical chemical tendency is the same, but bacterial dynamics are different. So that's why as I said, I started the collaboration with Mirko Horkera from 20 years ago. Firstly, start from pathogen in the natural environment, but now moving to the harmful algal bloom in Chile. But now we are moving to next stages related to bioaerosols. In the case of bioaerosols, I have so many collaboration with, especially in Chile. For example, main partner is La Frontera University, but I have also collaboration with Magallanes University, Federico Santa Maria, and Los Lagos University. And also now I have collaboration with Spain, especially in Barcelona Research Institute, and now I have a collaboration with Slovenia also. So now becoming firstly start from only one-to-one collaboration, but now moving to next stage to the worldwide collaborations. But finally, I hope to say my sincere thanks to Horkera, because we met 20 years ago like this. He is so young at the time. I think I'm not so changed. Thank you for listening.
Thank you very much. It's not my plan, change, please. So we have time for a couple of questions. Lucas, please.
So thank you for the nice talk. I would have a question.
Yep.
Why is the percentage of these non-pathogenic mycobacteria so different between the US, Europe, and Japan? Has this to do with water treatment before it gets to the apartments, or what's the reason? Do you have any speculation on that?
Yeah, so that is a most important question. So that's why now I'm doing keep regular monitoring of the water using the because especially important point is not only percentage of the percentage difference in US and Japan. More important point for me is patient of caused by non-tuberculosis mycobacterium, NTM infection is highest in Japan compared to other world. So it means percentage is very low, but patient very high. That is the question for all the researchers related to this topic. So, but anyway, now currently, when I take a sample in the water plant, I always measure the alkaline, chlorine, ferric, or many other chemicals. And there is some correlation with mycobacterium concentration and other such chemical and some concentrations, some relationship, but not so high. And also, I took a sample from, for example, river water to precipitation, filtration, chlorine treatment. Every step I take a sample and I check the abundance of mycobacterium, but nothing is changed by the treatment. And the treatment is very similar to US or Europe.
Microbacteria or non-pathogenic microbacteria are very well degrading pollutants and micropollutants.
Yes.
And is there a correlation to the micropollutant level you have in the water?
So it means if it's true, Japan water is very, very clean, right? US water has so many pollutants.
It's a hypothesis.
Yeah. I never check about the concentration of pollutants. But percentage is very low, but patient is very high. So that is a big question. No one knows about that. But the important point is, to this time I don't say anything about that, but from the genomic epidemiology, the type of genome type of NTM, non-tuberculosis mycobacterium, is totally different between European type and American and Japanese patient type. Japanese type is very similar to Japan-Korea type, and European American type is totally different. So it means some environment factor can enrich only a specific genome type. Because NTM is a non-pathogenic, non-tuberculosis mycobacterium. It means Mycobacterium avium is the most famous one. Mycobacterium intracellular and several abscessus, Mycobacterium abscessus. These three are most famous non-NTM, non-tuberculosis mycobacterium, which has a pathogenicity. So these three are very famous in the cause the lung infectious disease, but percentage is highest in Japan. So I don't know what kind of factor can enrich such only a high virulent species. So no one check about the species level identification for the mycobacterium. Maybe in the case of US, mycobacterium genus percentage is very high, but maybe most species will be a non-pathogenic one. Japan is very low, but all pathogen, maybe. I don't know. We need to check about that.
Another question?
Congratulations for your mobile labs. I want to ask about it. Which variables this lab can measure on site?
Ah, I see. About the suitcase laboratory?
Yeah, the suitcase lab.
Yeah, because we have two types of suitcase laboratory. One is only for detect specific genes by LAMP methods.
On site?
Yeah, on site. Yeah, because first version of suitcase lab is not this one, smaller one, has a, for example, bucket for water sampling, and a device for filtration, and a device for extract DNA, and also a reagent for LAMP reaction to detect specific gene. This is an old version. Which one is already installed in his laboratory, I think. You have in your lab, right?
Okay.
And another one, new one, this one is now shipping to here. Now, I think in Santiago, I don't know. But yeah, maybe Santiago will stop 2 months.
Did you try it with CRISPR? CRISPR 12?
No, for this one. Because this one, new one is for analyzing bacteria or fungal community. Yeah, because it means that new one has a clean bench, mobile clean bench in the in the suitcase.
Great, thanks.
Can you speak about the inside, the outside, the outdoor, sorry? What happened with the air condition?
Ah.
It is extremely, the dangerous, I suppose.
Yes, so many air condition maker much care about the fungi from the air condition. And of course, there are so many different kind of fungi inside the air condition. So maybe that is one reason why inside and outside is different. So in the case of traditional Japanese house, they usually don't use any air condition system. So that's why I think.
Is it the difference between the
Yeah.
United States, for example?
Yeah, totally different.
Okay, thank you.
Another question? Okay. I had this session is about the transfer to the society, the science. Before the pandemic, you start to work in the aerobiology before pandemic, after pandemic. You know, in Chile, the most science financed by government. But in Japan, we had the government, also the industry, the company. In Japan, you don't have problem with the company put money for the science. Compared to Chile. Before pandemic, after pandemic, the government and the industry is worried about the built environment, the microbiology in the air on the side. Do you think it's a change now? Trying to design better product or better house, or is pandemic finished and don't worry again for the micros? Before pandemic, nobody worry about the air or the microorganisms in the built environment. After pandemic, during pandemic is very important. And now it's after pandemic, not so very important. How is the vision of the Japanese government and the Japanese industry about the built microbiology?
Yeah, I think that point is not so different before and after, between and post pandemic in Japanese cases. Of course, Japanese government tried to restrict many things, but for the normal Japanese people, the way of thinking is not so different because maybe some people know, only a few people know, always Japanese people wear masks before pandemic, before COVID, always wear masks all the time without any COVID or anything. So our way of thinking and our culture is not changed at all. But of course, the percentage of people wear mask increase. So that is a difference. Yeah, but other important point, wearing mask is different, no difference. But about the way of work is different because Japanese government try not to come, not to meet with other people, try to prohibit that. So it means many people work in their own house. So it means if usually people in Japan work in an office for 8 to 12 hours per day, but most people not allowed to go to the office, and also government try to prohibit to go to restaurant or go to work, go to get to some transportation systems. It means that so many people try to stay in their houses. So it means our own house environment is becoming more important. So that's why around this condition, air condition maker and some such kind of sterilizer maker and other housing maker try to sell more and more product to purify the air. And some, for example, in the case of even a washing machine for dish or clothes, now we have a machine with UV lamp for normal washing machine for dishes. It's a ridiculous, I think, but many people hope to have such kind of UV sterilization system to everywhere. Yes, so that's an interesting point. But for the researcher, government also prepare a new grant to improve the COVID researches. But in the case of Japan, both private sector and governmental sector equally prepared the new grant for COVID researches. So not so different. Yes.
Okay, thank you. Few applause to. Thank you very much. We invite to the third speaker. His name is Carl Isaac. Carl Isaac belongs to the La Frontera University. They explained how to connect the science with the society, with the company too. That's a very interesting point.
Eso. Voy a poner dos cosas abiertas.
Okay, the title of the presentation is Science 2030 is New Opportunity to Connect the Laboratory with the Market.
Okay, my presentation needs to be in English. I don't know. I can make it in Spanish. No problem. Okay, okay. There are English people here, people who only understand English. Okay, great, great, no problem. I will try my best. Okay, this is not a presentation. This is some kind of conversation. It's more relaxed than a presentation. I'm Carlos Isaac from the Institute of Innovation and Entrepreneurship of the university. And I worked many years in innovation and technology transference. Then I want to talk about some things I learned about more than 27 years working on that. And a project that we have now that is Science 2030. We have a project with 6 universities of Chile trying to connect the science with the industry. Okay? How can we join these two worlds? How can we make the university most innovative, most entrepreneur, to push from the university to the industry, to the society, to solve the big problems, the main problems that we have in the world at now? And a question, mainly for the students. in which state will you be in 10 years from now? You can take your mobiles, take a picture to the QR code, and respond. The students, all the people who are students, PhD students. In which state you see yourself in the next 10 years? Not too much students? Not too much PhD students? No, I am asking the students. Yes, but they need to take the picture and make some response. I will see if someone is asking the question. Okay. Cooking in my restaurant, working. Working. Nobody else? Okay, this is not function. I will go to this, the usual way. No? No, it's working. It's working. The people are responding. Okay? Researching, working, some investigation laboratory in UFRO. Someone is in entrepreneurship, science entrepreneurship. Someone is trying to work in the industry. Okay? Let's see what's happened here in Chile with the work for the PhD students. And I put these two graphs. The left one is Chile, the right side is USA. Okay? In Chile, a good news is there are no unemployment in the PhD students. Most of you will have work. And if you are in States, it's the same. Most of the people who have a PhD have work. This is a good news. A second thing, in which area you will work? We compare here United States and Chile. In the United States, most of the people work in the industry and the government. And in Chile, almost 90% work in the university. I will go for my glasses because I'm a little old and I need glasses. Okay, then we have a huge space here. Yes, we have a huge space here to work with the... Marilu, ¿vas a ir a avisar eso? Ya, sí. I prefer to wear. And where are they? Presionar. Presionar H. I need to put my glasses. Ahí está. No estoy resolviendo ningún problema. Seguridad alimentaria, reemplazar plásticos de origen fósil. Mira cómo van saliendo. La eficiencia de los metabolitos contra la inflamación del cuerpo. descontaminación de agua, contrarrestar estrés oxidativo en plantas, waste management. Ok. ¿Por qué pregunto esto? Porque quiero conversar con ustedes. Why I'm asking about this? Because I want to talk about the innovation process. The innovation is the process that can connect us with the industry, with the consumers, with the problems we need to solve. And I want to show you a very simple model, what we did with Erco Auto of Imperial College many years ago. And it's the model is different from Canvas. Because Canva, you know Canva? Many people work with Canva, but Canva is very, very focused in the market, in the client. And we work with this model, and this model not only have the market pull, but have the technology push too. Because it's not only about the consumer. When you work in technology, when you work in science, you need big investments. And this investment, you can change it every time the client or the consumer want a different thing. How you build some kind of framework to give the consumer what they want, but have some backup to change if it's necessary, to scale, and to learn. Is this what I'm trying to show you in a little slice? Then, the first things is not only about the market, because the market changes every time. We, as scientists, we need to stay a little beside of the hurry of the market. We need to see it from little far away, okay? And think about the market pull, okay, but from the technology push too. First, if we think about the necessity, the needs, we need to look not at now, some years ahead. Two things. What are the technology trends? And what are the big problems that we face? Okay? This is not asking a survey to a consumer. This is the capacity to look beyond. Okay?
A vision.
It's a vision. Yes, it's a vision. Because in science, all the things takes time. Then you need to be at first looking there, not here, there. Okay? For example, which are the trends in technology? AI, nanotechnologies, CRISPR, metaverse, cloud computing. Which are the technologies that will be part of our future development? Which are the main problems? Water supply, energy, health. Not the specific problem, the big picture. Okay? This is the first thing. Not to look what the customer wants now, but the trends in technology and the trends in the problems that we will face, or the problems that we are facing. Okay? This is the first. Just look this video. Maybe most of you know about a company named Notco. You know Notco? Let's see a little video. Did it video? No. The video is not functioning. Okay? Then I will explain that Notco is a company that uses AI to produce food. Okay? But which kind of food? Vegan food. Why? because the people do not want to harm the animals. They are seeing this trend, the trend that people don't want to harm animals, that global warming is produced not only by, enhanced by the animals, for example, because the cows produce methane. Then these companies see these trends in consuming. And these companies see the trends in AI. And they develop food using AI to create food that is exactly that the real food. As same as the real milk, as the same as the real mayonnaise, as the same as the real ice cream and the meat. They use AI to match, they match AI with the consumer trends. Yeah, they go on. And they now have more than 50, the last time I know about it, they have more than 15% of the market with this company. Second thing, this company have a value proposition, very specific, very focused. They produce food built with vegetables, only vegetables. That's all. This is the value proposition, okay? And what is very important, they have a specific knowledge to do that. And I want to stop here talking about the specific knowledge. The specific knowledge is something that you cannot learn on a book, on a paper. The specific knowledge is something that you create in your lab. The experience, the experiments, the data set that you create over the years, the knowledge that is not in books and on papers. You create the specific knowledge. We need to create our own specific knowledge to enforce this task. Someday I meet with them in a lunch, and they told me that in their doctoral thesis, they have a lot of enzymes and food specifications, a big database about food specifications and components and nutrients. And they know every component, the viscosity, the smell. They do a great work targeting all the ingredients and make it a huge database to train the intelligence system. This is a specific knowledge. There's no book about that. They create it in the lab. This is very important for the value proposition. And I want you to think about it now. What is the specific knowledge that you are creating in your lab? This knowledge that only you know about it. Which knowledge you are creating? Which database? Which set of experiments are you creating? That will be very valuable in the future. Very valuable, because only you and your team have it. And is the result of years of effort and experiments and data. Do you have it? It's only a question to think about it. Okay? Then we see the needs. We saw the value proposition. The market is not the opportunity. The opportunity is when the market matches with your value proposition. The market is not for everyone. The market needs to match with your own capabilities. Need to match with your specific knowledge to have the advantage. When Paul told us an hour ago about his patent, and the question the company is asking, are you sure no one will copy this patent? Are you sure the Chinese can copy this patent? Are you, why you do not produce this more cheap? Many questions about this real opportunity, because to have the product or to see the market is not an opportunity. It's the match between them that is the opportunity. Okay? And then I will talk a little about the architecture. For me, the most important part that we need to be aware when we are scientists or scientists trying to connect with the industry. And in the architectural part, we need to have in mind three things: scale, scope, and learn. What we are producing, what we are doing in our lab, is it scalable? What we are doing, what we are producing, have a scope? And there's a difference between scale and scope. Because scale is to produce many of these. And scope is use the same product to be useful for different things. This is a scope. For example, Dr. Paul's enzyme, protein, what do you have? A compound. Dr. Paul compound. It can be useful for sunscreen. Great. It is useful for using shapes. It's useful to, for example, to drink. The same molecule. Can use this same molecule, mix it with something else in another kind of use. This is the scope. Understand? When we build something that is not only useful for one thing, but maybe for different things. And the last one is the learning capability. Are we learning fast? Are we learning cheap in our laboratory? Because the ultimate competence is to learn more fast and more cheap than the competence. How can we learn? And now, the data. Use a lot of data. The data make us learn. Are we using machine learning? We have data scientists in our laboratories trying to pick up the results and learn fast from it. We need to think in these three things to build the architecture of our laboratory or our challenge that we face. And the last one is the leverage capability. Many times, people ask me, I don't have the money. No one give me the money to build this huge building that I need, this big equipment that I need. And they think from here to there, straight in the first step, you need to cut the elephant in pieces, okay? And you need to eat each piece at a time. But it's not only to cut the elephant, because each slice is a different size of the next one. Not all the slices are the same size. At first, you try little bites, but you need to have results. You need to have real value from your first experiments, from your first projects. Make a cheap project and demonstrate something. Fix a goal and have it. And when you have a goal, when you have something valuable, even if it's very simple, you have something. And from this, build the next step. And go for the second project. At first, 1 million project. Everyone give you 1 million. It's not difficult. You need to find what I need to research with 1 million. Something very specific. And then, got it. When you got it, you have credibility. You have learning. You have more skills. You go for the next. 20 million. 20 million. And with the second 20 million, you need to obtain a second objective, very specific. It's not about giving sticks everywhere. It's about going for specific aims each time. Specific aims. You need to build it that way. And then the leverage is produced because the system always works like an exponential curve. But you need to understand that you need a goal here, you need a goal here, and you need a goal here. Very specific. Step by step. And the third one, in architecture, okay? The first one was the scale, scope, learning. The second one was the leverage. And the third one is the modularity of our actions. The thing that I'm doing now is useful for other kind of research, for other kind of challenge. When we think about our laboratories, we are thinking about this, something like this Rubik cube, that from the same parts, we can have different kind of projects using the same models. It's not about to build a project and then when we build the next one, we need to build everything from the scratch. You need to think which components, which process, which way of working you will build, that you can interchange them to build different things. These are the three main things we think about the architecture. And there are companies that are doing this now. For example, Moderna. Most of us have a shot, a vaccine shot with Moderna. Okay? Look at how many different vaccines Moderna have for different diseases. Many diseases, different. Because Moderna used a lot of data analytics, a lot of software, and they have a very modular system to work. And the same protocols that they use in one vaccine are useful for another vaccines. Look how they can build a wide range of vaccines from one company, but using cloud, Internet of Things, AI, and analytics. They are making the lab, the biological lab, to be a software lab at the same time. This is the idea of the architecture. How can we scale, make scope, and learn fast from our laboratories? Okay, this is a preview because what I tried to talk about is about Science 2030 project. All the things are involved in the Science 2030 projects. We try to match the science with the companies and with the university. Okay? We are working in new courses. For example, today in the University of La Frontera, we have courses on AI for science and courses of CRISPR. Okay? You know CRISPR? Many of you? I know. The scissors. Okay? That's the reason I asked you about if you can use CRISPR for your mobile labs. Because using CRISPR-12, I think is very easy to detect bacteria and fungi. And we have many courses. And now is Carol? Carol, are you there? No. I saw Carol minutes ago. Yeah, Carol is teaching a course on CRISPR right now, our third course this year. And in January, we will have a course on AI. In January, we will have a course on AI for PhD students and postdoctorals. Okay? For you to know that it's coming. We had one last year, and now we will have another course on AI. It's very easy to use clouds tools for AI. Believe me, it's not rocket science. It's very useful. And now all of you can use it for your own labs and your own experiments. Okay? Okay, no, I will send it. We did many workshops. We did 3 workshops this year. One of them with this marvelous woman. This was the Nature Woman of the Year. And we had it in the university in a very interesting speech talking about CRISPR and electronics. She designed microchips to detect CRISPR straight on the computer. We had a hackathon that we finished with the 6th university and the University of La Frontera, and the students of the University of La Frontera won the hackathon. And then they went to Punta Arenas and Tierra del Fuego, and they have now resources to put this idea in a project. 2 weeks ago, we have a program, Deep Tech Acceleration Bootcamp with professor of Imperial College. It was a very successful activity. 35 PhD students and some of them graduate students. And now we are finishing our Lance concourse. It's a grant to go abroad with the thesis advisor to solve global challenge. Okay? And maybe we will have the results of the concourse in few weeks. Okay. Be aware about our artificial intelligence course on January. And this is the kind of things we are doing in Science 2030 to complement the science in the university, to make the university most innovative, to make the people with an innovation mindset. Okay? And we are working on that with these programs and many others that we have over the year. Thank you.
Thank you very much for the presentation. We have time for questions before the coffee break.
This year, we have a course named GT, Management of Emerging Technologies, and we have something like 25, 26 students. All of them great, great students. I see every year better students in innovation. Not in science, but in innovation, they are very worried about it.
Thank you.
So, first of all, thank you for doing your talk in English. It's very helpful to me. I wouldn't have gotten nearly as much out of it. I would have gotten a little bit, but not much. So you had a lot of slides from my previous employer, the National Science Foundation, on the demographics of American scientists. And I just want to caution you. The United States is quite different than Chile. First of all, we have an essentially unlimited number of universities, many of whom in the 1990s decided to give PhDs, but they're not Research One universities. So when you design an ideal for how universities work with industry, you can't give away your seed corn, which is your university professors who work on the same aspect of biology for year after year after year and become expert in it. And I can tell you that companies like Bayer and previously Monsanto pay me thousands of dollars a day to come and talk to them because they don't have anyone that they pay that to. But they have lots of scientists. And their scientists work for 1 or 2 years on a very specific project. So they need the universities to feed that knowledge into the whole system. I could go on for a long time on this, but I won't. So a better model for a country like Chile would be something like Sweden or Norway, where they have a relatively small number of faculty in a particular area, and they have a robust industry as well. And if you wanted a model that would be more directly applicable, not that there's anything wrong with the United States, I live there, and now we no longer have a president that I was embarrassed by. The concept, however, is quite different because of our demographic. And it's a demographic that...
You have 4.5 million PhDs.
Right, and then I just wanted to put one other little minor thing, which is you talked about the hierarchy of funding, and I work for that funding agency, so I take it properly. It is true that you need to take it a step at a time, but one thing that a developing science needs is to remember that you always want to hold a carrot out that says, for that very special, unique idea, you can get a lot of money.
Yes.
It might not be a lot of people getting a lot of money.
But you are not a younger one trying to ask for that money.
No. But as a younger one, I used to be young, believe it or not.
Me too.
But for the younger one, you want to say, as you work and you get something really good, maybe, just maybe, it'll be super good, and somebody will give you an opportunity to really do it, And so, however you set up hierarchies, in plant sciences in the United States, when I started, you had no opportunity to get a big grant. But the genomics era changed that, So now I have multi-million-dollar grants. I wish I had more, but you can now get multi-million-dollar grants if you have that good idea. And that has changed my whole life, because now I look, and yes, I have my basic grant, which is my creativity grant, which isn't big, it's not small by Chilean standards, but it's not big. But once in a while, I get something really good, and I can actually compete for more money. And that changes my whole perspective in life. Right? I just want to hold that out, that you don't want to say just gradually, because if you get something really good, you would like to have a government or whoever funds say, yes, do it. Right?
Yes.
And it doesn't have to be very many people. It just has to be out there as a carrot that says maybe.
What I'm trying to expose is the concept of leverage. Okay?
Yeah.
because I'm talking about my own experience.
Right, I'm not disagreeing with you. I was only amplifying.
Amplifying, great. No, because I saw many researchers. In 2005, one researcher asked me for 1 million pesos. And with this small amount, he built a bigger project and have many data from this new project. And then they asked for a big one, and finally he managed 5 million, and the government give him 5 million, and he start with 1 million, because they make leverage, because each project connect him with companies, businessmen, more data, more results, and then the system go up this way. But sure, someday you will need big money over the table. And you will need to be prepared for that. It's better you need to be prepared.
We never give beginning investigators all of their money of a 4-year grant in the first year, because experience tells us that some beginning investigators spend all 4 years in the first year.
Yes, yes, sure. I'm trying to challenge you. Not you, but you. Okay. From the young ones. A question. A question. It's all clear. Make leverage on your life. Leverage is very important thing. In life, many things go in compound rate. And for the compound to function, you need time. It's the same as the bank account. You need to put your money on the bank account and not take off in 5 years. Leave it there, and you will see, finally, will go up. Okay, thanks.
Okay, thank you very much. Now we had a coffee break. We had the opportunity also to make a question to the speaker, okay? To have additional question.